which have comparable frequencies. Is that so?
No. First part is right. I have 2 order parameters, for your language, 2 collective variables. That part is correct. Second part, no, we had no inkling what kind of number we'll get. And this, we spend a lot of time getting these numbers. And I told you, we already have, I at least already had enough past experience with these numbers. Now, I mentioned that the reactant well of the insulin dimer in its native state is very broad. Now, and I expected in the Y direction much smaller barrier frequency, but fitting, well frequency. So that is not here, unfortunately, but yeah, it is here, 6.13. So omega A, first one, which is the WS, which for some reason it's blue. That's what I didn't see it. So this is 7.68. So this is in this field a reasonably big difference of 15%. Okay. So omega Y is flat. More flat you have is the bad news for the reaction, because you have a better entropic trap, because the partition function becomes large. So this is an important point. Barrier frequencies, usually you depend very strongly on the activation barrier, and we have a very large activation barrier. This is one of the reasons we have a barrier frequency in the X direction, which is remember, barrier frequency in the reaction coordinate is like this, convex, but the one in a trapping Y is harmonic frequency, right? It's inverted parabola, one is parabola. So they are, this is why they are certainly uncertain. I think I'll vouch for omega reactant well. The barrier frequencies require a lot more care and work, but we did it by 2 ways. One by fitting as and Chandrasekhar kind of things, but you have to see these are in one direction, it's a unstable mode. An unstable mode in a short time you can do, but then these Chandrasekhar's equations become cos, as I said. That's the one way. Another way, just direct fitting. We have done both the 2 ways, and with the similar results. This is to be, this parameter space, I am sure, need to be looked into much more carefully. Given that the rate that we have found, if we discard 1D TST to begin with and 1D Smolakowski to begin with, because no reason, then rest of the things are not too distinct from each other. If you don't take TST results seriously, because they are not, because it's pretty damped, because you have these things which are moving and you can see that they are really, water acts at a distance and gives rise to friction and vibrates. This is a beautiful thing that we are developing now. So I have, if I get rid of those things, which I know obviously wrong, I have a fairly consistent picture, which means that there might be cancellation of error going on. I know you remember we gave a talk where also all these things kind of gave the similar rates in your different collective variables. So there may be certain some saving grace is there. A lot of entropy is here that we have not looked into, but I would like to look into a little bit more carefully, like the reactant well. So yes, there is still a lot to worry about and a lot to think about.
Thank you.
So I think in the interest of time, we'll have to stop there. So please join me in thanking Biman for a wonderful talk. And Biman, there are a couple more questions from Harish Vashith, and I believe also Aaron Dinner in the chat. If you're available to answer them in the chat, that'd be terrific. So, so thank you very much, Biman.
Thanks a lot. Bye.
So our next speaker is Gerhard Palmer, and so as Gerhard is getting set up, let me introduce him. So Gerhard is a director and scientific member at the Max Planck Institute of Biophysics. He received his PhD from the University of Vienna, and he has a diversity of interests, as we all know, in quantum, all-atom, and coarse-grained modeling of biomolecular structure, motion and function, supramolecular assembly, membrane channel and transporter function, hydrophobic effect, electrostatics, and of course, methods development and collective variable discovery, including equation-free and path sampling methods. Gerhard has over 48,000 citations and a large number of awards. I'll just mention a few. He's a fellow of the American Physical Society. He won the Nancy Nossel Scientific Mentorship Award at the NIH. And in 2010, he was the Raymond and Beverly Sackler International Prize winner in biophysics. So we're very privileged to have Gerhard here, and so I won't take up any more of his time. And so Gerhard, please.
Well, thanks, Andy, and thanks all the organizers. I hope you can hear me. This is a real pleasure and an honor to be part of this meeting. And it's, however, also quite sad, I think, for all of us that we're not together in India and having this celebration of science. Instead, we're trapped in our offices around the world. So, which also means, yeah, good evening to India and good morning to the US, and I guess a good day to Europe. So I will be talking about some recent work, which is a collaboration with Alex Noy's group at Lawrence Livermore on the delivery of drugs with carbon nanotubes. And but starting out, I will give just a brief history of my interest in carbon nanotubes, and then lead over to the nanotubes as delivery vehicles. And at the end, I will give a bit of an overview and an outlook of what we are doing right now, also to give you a taste of where we are heading. So I think, as some of you know, carbon nanotubes have been a bit of an obsession of mine for now 2 decades. And this started with Jay Razaya visiting the NIH for a summer, and we being interested in hydrophobic effect. And one of the observations that we made was that small carbon nanotubes are actually quite compatible with water and can provide conduits for high speed water transfer. So they're really interesting nanofluidics conduits. But they were also good conduits for the transfer of protons, and this came from a collaboration with Christoph Delago, where we found that a nearly perfect hopping mechanism was in place and allowed really very fast proton diffusion in excess of, I think, about 40 times that of the diffusivity of protons in bulk water. So it was a really quite remarkable medium. But nanotubes, of course, have attracted the interest of experimentalists and also not just computational people, and they have been studied very extensively for various applications. And actually, Alex Noy was one of the first, together with Olgica Bakayin in one team, and then Bruce Hines in another team, to show that these fast water transfers that we had seen in our MD simulations actually did occur in real systems. And so Alex Noy also caught the nanotube bug, has been studying them for a very long time. And a few years ago, they succeeded in getting nanotubes inserted into lipid bilayers, which is, of course, something that I think many of us have dreamed of as a possible application. And they could show that you can transfer ions and protons across these membrane inserted nanotubes. But another thing that they observed that I thought, and my group thought, was really very interesting was in their electron micrographs of lipid vesicles with nanotubes, that sometimes the nanotubes were actually stapling bilayers, different bilayers together. And so you had these, as shown here on the left, 2 vesicles brought in close spatial proximity with a bridging carbon nanotube. So one of the questions that we then raised in our discussions here in the group was whether these nanotubes could actually not just staple the vesicles, but bring them to fusion. And that of course, is very interesting biologically because fusion plays such an important role in so many biological processes. And I'm not going to go through them, but one that I think we are all currently dealing with is, of course, viral entry at the top, but also the delivery of vaccines into cells can be achieved through types of fusion processes. So in the cell, of course, membranes have to fuse in the great wide range of cellular dynamics. So fusion, however, as I think, again, most of the people here will know, is a very slow process because lipid bilayers tend to repel each other, partly because of electrostatics, and also for other reasons. So they're already difficult to bring into close spatial proximity. And even if they are close in space, a quite large activation barrier has to be overcome to get them into fusion, because you have to open up the hydrophobic core of the two bilayers and bring the two cores into contact for the two leaflets, initially four leaflets to fuse into a new single membrane structure. And that leads to timescales of fusion even in like very densely packed membrane systems studied here in the Rothman and Pincé labs on the timescales of tens of minutes, and barriers on the order of 30 kT, so very, very slow. And that's why biological systems have evolved rather elaborate and intricate machineries to facilitate membrane fusion. And the most well-studied machineries in this regard are stair proteins. But these days, we're of course also all learning about the spike protein, whose primary function indeed it is to get the viral envelope to fuse with the host membranes, either the plasma membrane or the endocytic membrane. So the question that we asked was whether carbon nanotubes are actually candidates as possible fusogens, so a synthetic fusion machineries. And to study that, already a few years ago, Stephanie Linker and Ram Bhaskara set up molecular dynamics simulations of various membrane systems using the Martini coarse grain simulation model. And in these simulations, they used vesicles, small lipid bilayers, and tethered them with carbon nanotubes. And here you see one of these simulations. So the water is not shown for clarity. The golden spheres are the phosphate groups of the lipids, and only the green tails of lipids in direct contact with the carbon nanotube are shown. So you can see that quite quickly, the outer leaflets of the two vesicles were pulled together, and that led to outer leaflet fusion. And then over a somewhat longer time scale, you have this hourglass intermediate structure, but at about this moment, it's this remaining diaphragm at the center popped, and we have a complete fusion with a now peanut shaped final fusion product. Here you see snapshots along this process that's seen in the simulations by Stephanie Linker, where you see an important part is that the carbon nanotube pulls the vesicles together and tilts, and over this few hundred nanosecond time scale, fusion happens by establishing contact between the tails of the inner leaflet lipids, shown here in gold and red. And here you see this really central structure in this fusion process. This is the hourglass fusion intermediate. And what to look for is shown here at the end of the movie is the intimate contact between the red tails of lipids from the inner leaflet of the left vesicle and the yellow tails of the lipids of the inner leaflet of the right vesicle. Because in the fusion process, these two lipids have to fold over and open the diffusion pore by breaking the symmetry of this structure. So what was reassuring to us was that fusion at essentially constant volume, as you would expect for membranes that are impermeable to salt, results naturally in elongated tubular or peanut-shaped structures. And these structures turn out to be abundant in the electron micrographs shown by the Noy et al. Paper. So we were quite confident that we were on track to something meaningful here, that our simulations were actually capturing real fusion events that led to these otherwise unexpected elongated structures in the experiment. However, when we then reached out to the Noy group, turns out they had been, of course, thinking about this problem themselves for a while, but they could never get their experiments to produce fully convincing experimental support of this notion of carbon nanotube mediated membrane fusion. But they didn't give up, and they persisted, and over the last 2 years or so, actually now maybe closer to 3 years, they accumulated enough evidence that clearly showed this carbon nanotube mediated membrane fusion is real and is possible. And just very briefly here, the basic idea of the experiment is to load a lipid vesicle with a fluorescent dye. In this case, this is a self-quenching dye. That means that above a certain concentration, when these molecules are quite close in space, fluorescence is suppressed by the interactions between the dyes. Fusion with unlabeled vesicle leads to dilution, and this thinning of the dye removes to some degree the self-quenching effect, and so fluorescence then appears. And when they monitored this, they could clearly see in a dose-dependent manner, depending on the amount of nanotubes, that they could see fusion. And in the control experiment, that is carbon nanotube without, vesicles without nanotubes, they actually see an opposite signal. There's just the usual bleaching effect. So this is was quite strong evidence that fusion happened, but there's even stronger evidence by then using in addition dyes that are now in the lumen of the vesicles. And so when you then fuse with unlabeled vesicles, again, you can detect the change in the fluorescence and confirm that you have not just fusion of the lipid bilayers, but you actually have exchange of the volume of the two vesicles. You have actual fusion with an open diaphragm between the fusing membranes. But then something interesting came out of the experiment. So Injia Ho and Alex Noy did careful kinetic analyses, collected data, quite extensive data to do this, and plotted the rate of fusion, or in this case, the half time for fusion, as a function of the number of carbon nanotubes per vesicle. And what they found was it was not the expected 1/n dependence, but a 1/n squared dependence, which, of course, to kineticists indicates that the catalytic species, the fusogen, might actually require some form of interaction between the carbon nanotubes. So and this is where molecular simulations again came in to give us an idea of what that species could be. So Mark Sigel, in the meantime, joined my group to work on asymmetric membranes, and actually has just graduated, received his PhD. So Mark started to set up simulations of monomeric carbon nanotubes as fusogens, dimeric carbon nanotubes, where you have the two nanotubes lined up in parallel, side by side, and trimeric nanotubes, where you have them arranged like in a little triangle. And what he found, and this is shown here in the movie, and I hope you can see them, is that dimers fused the lipid bilayers very, very effectively. Monomers and trimers were not very powerful as fusogens, and we could modulate this fusion tendency by changing the barrier to fusion by using asymmetry in the number of inner leaflet and outer leaflet lipids as an additional control parameter. And it was quite clear that in the conditions where fusion is slow, kinetically not very facile, the dimer, where the catalyst or the fusogen is really needed, the dimer turned out to be by far the most powerful fusogen possible. So why is that? So I've already shown you based on Stefanie Linker's and Ram Bhaskara's work, what the basic principles are of fusion, but I will now go into this in a bit more detail. So what you see here is this a zoom in on the connection between the two fusing vesicles in top and bottom, again in the gold red coloring for the two inner leaflets, and with the tails indicated for lipids that are in contact with the carbon nanotube, and those involved in the actual fusion process indicated as thick sticks. So what needs to happen is two things. First is the fusogen needs to be able to pull the two vesicles into close spatial proximity, so close that can rupture the outer leaflets and bring them to fuse. For that, you need substantial hydrophobic surface such that the vesicles want to be wetted by the lipid tails. The second problem, however, is that what I already alluded to, the breaking of the symmetry, that on one side of the nanotube or on the other side of the nanotube, the lipids have to swing over. These are the inner leaflet lipids shown here in red and yellow, and push the nanotube to the side and establish inner leaflet contact. That's when the central pore forms and fusion completes. And it turns out that thin carbon nanotubes in a dimer form provide the perfect trade-off for this process. They have enough hydrophobic surface on the flat side, but only a narrow space to bridge on their narrow side. So that means we have a strong pull and an easy crossover and symmetry breaking for fusion to occur. That was very reassuring, but the question then was, can you make something useful out of this? And of course, one idea to use this is in drug delivery, using loaded vesicles as carriers of high-value drugs or very toxic drugs that would otherwise cause damage to the cell, such as in cancer therapy. And that's what Njia set out to study. So she prepared such vesicles with a cancer drug, doxorubicin, and then studied their effect in cell culture. And what you can see is in this plot of cell survival is that there is no effect if you just have vesicles with carbon nanotubes. So fusion itself is not toxic. If you have just carbon nanotube, that's also not toxic. There's no effect. If you have the vesicles loaded with doxorubicin, but no carbon nanotubes, there's also no effect. Same thing is no effect with only buffer. However, if you add free doxorubicin, this is the second from the right, or doxorubicin, doxorubicin loaded nanotubes, vesicles with carbon nanotubes, you have a very drastic drop in cell survival. So this actually works. And what is very nice and encouraging about this is that these vesicles fuse directly with the plasma membrane, so there's no risk of loss or chemical damage for the delivered substrate when it is taken up on the endocytic pathway. And what is also nice is that the amount, I'm just saying this briefly, the amount of drug that has to be put into vesicles when it is delivered in this way, whereas what you would need for a normal systemic delivery is really quite dramatically different. So this is really this vesicle-based delivery is a potentially very promising approach. With that, I would like to conclude the carbon nanotube part and take the remaining few minutes to very briefly to give you an idea of where we are heading in our group and where I see emerging challenges. So my institute, my group, and many of my colleagues are increasingly pushing towards molecular simulations of cell scale processes. And I think, and I'm actually quite confident that our tools and the computational power are now here to make this possible. And this is one illustration of this. This is a simulation done by Søren von Bülow, a PhD student who is now just about to graduate, of proteins at concentrations of about 200 milligrams per milliliter. These are all atom simulations with hundreds of proteins. And what he found, just can give you a vignette of this, is that when you look at the materials properties of these concentrated protein solutions, for instance, in terms of shear viscosity or in diffusivity, he was in agreement, his simulations were in agreement with experiments over the entire concentration regime, all the way up to the densities you would expect inside a living cell. And this is particularly reassuring because the reason for this high viscosity, about 3 to 4 times that of water in some of the systems, is the weak tendency for proteins to form transient non-specific interactions. And so our force fields are indeed sufficiently balanced to capture this, not just the simple hydrodynamic properties. Another area that we are very active and actively working on is the formation of so-called membraneless organelles, or protein condensates, or more generally, also condensates involving nucleic acids. And this is some work done by a visiting master student, Zakaria Benayat, who has been studying the self-assembly of diffuse low complexity domain into little condensates, droplets, and then extracting again materials properties for them, for instance, the low surface tension. But we're also looking at larger cellular structures now with atomistic simulations, such as cell-cell junctions. So for instance, here you see an atomistic simulation of desmosome, which is a cell-cell junction holding together cells and tissues under high mechanical stress, for instance, in the skin. And extracting from these types of simulations, not just structural properties, but really mechanical properties, such as the resistance to compressive and shear forces, and comparing them to experiment. And what we could explain as is work of Matt Sikora is, together with Achilleas Frangakis, who did the cryo-EM tomography experiments and also, of course, contributed much to the modeling of these structures, is that this truss-like interlocked architecture is the key for the superb mechanostability of these cell junctions. The last system I want to highlight, and I think that this is really something that personally I've been interested in for a very long time. I've been following the literature for a very long time, but I really did not expect that in my scientific lifetime I would ever be able to simulate a system of this complexity inside. This is the nuclear pore complex, which in humans is assembled from about 1,000 proteins, and has been studied by my colleague Martin Beck here at the Institute using high-resolution electron tomography. But thanks to the advances on the experimental side, getting ever higher resolution, and more recently on the AI-based modeling of component structures, AlphaFold, plus of course decades of crystallography and cryo-EM studies of the component, we could assemble a simulation model that of the ring structures in the nuclear pore complex, and study its dynamics and its coupling to the membrane environment using atomistic, sorry, not using in this case Martini coarse grain simulations, but I think that atomistic simulations are probably certainly possible in not too distant the future. So simulations were done by Mark Sigel, and the modeling of the structure on which we very heavily relied was done by Agnieszka Obarska, with input also from Jan Kozinski's group at EMBL. So the last point that I would like to highlight just very briefly, and I think this leads also over to the rest of the day and maybe the meeting, is the importance of AI, not just in modeling structures, but increasingly in driving our simulations. But this is something that we are also working very actively on in developing this case transition path based samplers that simultaneously sample slow reactive events and extract the relevant degrees of freedom and condense them into analytical forms. And this is work by Hendrik Jung and Roberto Covino. And with that, if slides move forward, I am at the end. I should thank my collaborators. I already mentioned on the left, I think everybody except Shekhar Garde has been very instrumental also in collaborations on carbon nanotubes and many, many more things. On the right, people who have been in my group, I should maybe mention in addition, Jürgen Kövinger has been involved in essentially all these projects, and Martin Vögele was a graduate student here in his modelings of carbon nanotubes. With that, I stop and take questions. Thank you.
Great. Thank you so much, Gerhard, for a beautiful talk. Please join me in thank you now, Gerhard, for being with us this morning, or this afternoon, or this evening, depending on where you are. And so we have time for a few questions. And so maybe I can start with a quick question. I was really intrigued by these peanut-shaped sort of fusion complexes that seem to form. In the absence of is the carbon nanotube maintaining this ellipsoidal geometry, and in the absence of the nanotube, would they become spherical, or is this sort of a natural state?
Yeah, so this is because so the membranes are water permeable, but not ion permeable. So when you fuse two vesicles in an osmolyte environment, such as salt, the volume is effectively conserved. There is very little in exchange of volumes. You end up with a structure where you have conserved area. Membranes are not very extensible, and you have a conserved luminal volume because intake or release of water would lead to osmotic imbalances. And as a result of that, fusion of vesicles that do not contain ion permeable elements lead to these tubular shapes. It's a little complicated because if you fuse more than 3 vesicles, then a red blood cell shape would become stable, but that has a high kinetic barrier. So it's not the nanotubes. It's a generic aspect of volume conservation.
Got it, terrific. Thank you. So a question from the chat from Jagannath. You've shown all these fusions via sort of a monomer, a dimer, a trimer. What is the result of the control experiment in the absence of a CNT? Will you actually see fusion eventually in the absence of a CNT or not?
Yeah, so we have not tried that. Other people, for instance, the group Miller group and the Rissalada group, they have looked into that, and this is, of course, it's a very, very slow process in simulations. they have estimated I think the intrinsic rate, and it comes out to be very slow. I do not remember the number for similar systems similar to us. What happens there is the dominant pathway seems to be that one of the lipid has one of its acyl chains flip out of the membrane, bridge over to the other membrane, and insert into the other membrane. So you end up with a splayed lipid, and that's kind of the transition state. And that's, of course, a very rare event, very slow.
Great, thank you. Another question from the chat from Prajesh Roy. And so he says, very interesting talk, thank you very much. And he was inquiring as to the dependence on the size of the lipids on the fusion process. Is there any critical lipid size necessary for fusion?
And this is a good question. I don't have, so we have played around a bit with size. Because there is, of course, a suspicion, thinking about these two processes of flipping over on the side and so on, that there could be a size dependence. I do not remember that we had any definitive results on that, but it was not something that we have, at least that I remember having explored. We have discussed it, but clearly your question is a reminder that we should look at that. We have used different lipids, but it was mostly variations in the head group region, like.
Great, thank you. Another question from the chat from Palab Dutta. Great work, thank you for the talk. Can you shed some light on whether diffusion of the carbon nanotubes may regulate the rate of fusion? Yeah, so again, another very interesting question. So what we see is that the carbon nanotubes diffuse quite rapidly within the lipid bilayers. That's actually something that Martin Vögele has studied in quite some detail. So, so that means the diffusion, so the localization, finding the right spot for fusion, I would expect is by itself not limiting the fusion process. Terrific, thank you. And then a final question from Satna Surbriya. And she says, what are the implications of the Cnts for transport of other molecules and also toxicity in the cells? Yes. So, so one of the interesting aspects, of course, is that carbon nanotubes are of a certain size will be permeable to all kinds of molecules. The ones that the nanotubes that we found to be the best fusogens are very small. They are, as it happens, pretty much the diameter of our original study of water permeation. So just wide enough for a single water molecule. This means they would exclude ions, all osmolytes. So that means effectively impermeable because the membrane itself is quite water permeable. And if needed, they could, of course, also be capped. Although I think that would be difficult because we want sufficient hydrophilicity at the end. So that's not an issue. Toxicity, of course, is worrisome, at least when one starts out, because carbon nanotubes remind of asbestos needles. However, the carbon nanotubes that we're dealing with here are incredibly small. They're very short. They're like 10 nanometer long-ish. And that there's studies on toxicity of small carbon nanotubes, even longer, like micron long, and they have, at least in these animal studies, seem to have very low toxicity and very rapid clearance. I seem to recall, we'll have to look this up, on hours through renal clearance. It's not an area that I'm an expert in, but the numbers look much better than I would have also naively expected. And that's also what we saw in cell culture. The nanotubes by themselves didn't seem to have a big effect, but clearly this is a concern for practical applications and will require very thorough studies. Terrific. So in the interest of time, please join me in thanking Gerhard again for being with us today for a wonderful talk. And let's move on to the next one. Thank you, Gerhard.
Thank you. Bye, everybody.
So our next talk before the break comes to us from Swati Bhattacharya. So as Swati's getting set up, let me introduce her. So she's an assistant professor at the Department of Chemical Engineering at IIT Bombay. She received her PhD from the Max Planck Institute for Polymer Research in Mainz. And prior to moving to IIT Bombay, she was an assistant professor of physics at IIT Guwahati. Swati's research interests are in the structure and dynamics of biomolecular systems, the development of algorithms and techniques to engage the timescale challenge in biomolecular simulations using kinetics-based enhanced sampling methods. And particular systems of application interesting to her include intrinsic immunity, intrinsically disordered proteins, and force spectroscopy. So, please go ahead, Swati.
Yeah, thank you very much. Am I audible?
Yes, we hear you well. Thank you.
Right. So thanks a lot. And first of all, thanks to Nishant and Jagannath for making this happen, and everyone else who were involved in planning this. So I'm going to be talking about scanning the entire conformational space of a biomolecule and trying to calculate the free energies. So the title can seem a little strange and even provocative, but my goal over here is to present a different approach, a local viewpoint to study conformations and their changes. And I'll try to walk you through the methods and also discuss some implications. So this work is in collaboration with Abhijit Chatterjee. Now, biomolecules such as proteins are shape shifters. In many cases, they can adopt multiple conformations, and there can be transitions between them, which are very important for functioning. So if you look at this schematic over here, I think I need a laser. Right. So if you look at the schematic over here, the individual points over here would represent atomistically resolved conformations. So all of these points would represent all the different conformations that a protein can adopt. And you can see these tiny little arrows over here. They represent the small moves that the protein can make. So as a protein moves, it is through these jigglings and wigglings of its internal coordinates. And when a small change is happening, it would usually involve only a few atoms, and this is what I'm calling a local move. For example, it can involve the changing of only one single dihedral. Now, large changes of a protein can happen through a series of small local moves. So as an example, if you look at a hinge opening that I saw recently in a MD simulations of a chaperone protein, and you can see this is a very big change in the system, but this hinge opening is essentially affected through a series of small changes, especially happening in this corner or the elbow. Now, if you want, you can collapse this entire transformation to a low dimensional manifold, provided you could define it, but that is not what I'm interested in. Rather, the question that I'm posing here is that, is there a way that we can predict all important conformations of the molecule, starting from a given structure? So what we want to do really is to determine the size of the conformation space, free energies and probabilities of the states. More specifically, we would like to at least discover and rank the top n most probable states, depending on the probabilities. And n can be a large number, can go to millions. So for this, we're going to focus on local degrees of freedom. So as my example system, I have this 20 residue mini protein, the TRPKH molecule. It has secondary structure elements and stabilized by some long range interactions. There's also a loop over here that can fold, bringing distant parts together. So can we take a local approach over here? Now, one important thing is that some form of enhanced sampling is going to be required, because if we just do simple MD on a folded structure, the only thing that would happen are these, tiny little twitches of the dihedrals, but it's really hard to see any major change happening. It's not going to spontaneously unfold in hundreds of nanoseconds, or even if I continue this for a few microseconds. So my preferred method over here is tiered MD, but I will come to that a little later. So the first thing is what do we mean by states, or what do we mean by local states? So in this study, I have ignored side chains completely and I'm focusing only on the backbone. And in fact, using just the phi and the psi dihedrals, we can build up the conformation of the peptide backbone. But in case there's somebody in the audience who does not work with biomolecules, let me put this a little bit more systematically. We need coordinates of all atoms in the backbone to reconstruct a structure, but instead of that, I can also work with a list of bond lengths, bond angles, and bond dihedrals, which is how we define the internal coordinates. But of course, most of you would know that bond lengths and angles don't really contribute to the multiplicity of the conformations. And of course, just for the sake of thoroughness, we actually verified it by computing histograms for every single bond length and bond angle and so on. They all turned out to be unimodal, which is really a feature of the force fields, I have to say. So the origin of the conformational diversity lies in the dihedrals, and more specifically, the phi and the psi dihedrals. There's also an omega dihedral, but that does not do anything. And thus, locally, the changes in the dihedrals are responsible for global conformational changes. So what in our framework, we define local states for every single backbone dihedrals separately. There are 38 of them in this particular examples, and for that we use our MD simulations. And as an example, I'm showing you here the histograms for two separate. One of them is a phi type dihedral, phi 13. One of them is phi 6, which is the 6th psi dihedrals. So this just goes to show that you can even visually identify multiple peaks, and these would correspond to different local states. And this is what we are doing for every single of these 38 dihedrals. So now, to represent the global conformation, all we need is just a list of these 38 numbers over here. And this is how we define our global conformations. Now, let's just assume something strange over here. Let us assume that all dihedrals are independent, which of course they're not. So my question is, okay, first of all, what is the number of local states that can be visited by any dihedral? And this plot over here on the right shows you that this number varies between 1 and 4. And there's only one case where we saw 5 dihedrals, but many of them are unimodal. They just show one state, and that means that they're quite rigid. Now, if we just multiply this thing, the number of accessed local states by each dihedral, the number that we get is actually the size of the total probable conformational state of the protein. Of course, many of those states would not exist because of self-intersection and many other reasons, but this is just the first example that I'm giving you of a large number that comes out of this kind of thinking. That number turns out to be 10 raised to the power 16, which is a huge number over here. So the message over here is that while the number of global conformations may be really enormous, if you look at individual dihedrals, they visit only a small number of states, just 2 or 3 at most. 4 is really the maximum in most cases. So, as I mentioned, plain simple MD really does not achieve much. So the route that I'm taking is to perform steered MD simulations with constant end-to-end distances. And in this case, we attached both ends to fixed anchors through harmonic springs. And we have set up multiple such simulations. Here you can see the movies of some of these over here. The question is, what happens to the dihedral values when we stretch the molecule? The interesting behavior that we've noticed over here is that the location of the peaks are not really changing. They're not shifting so much, but the heights or the probabilities associated are changing. So that means some local states are more likely at some stretching and less likely at others. And in some cases, completely new peaks are discovered as we stretch the molecule more. So in this second example, psi 4, it was practically unimodal at all reasonable end-to-end distances, but only at extremely high stretching, we saw that this peak practically vanished and we got a new peak over here. But another question is, what is the conformational diversity at these different stretchings, and how many states are we actually seeing in these SMD calculations? Now, my calculations were not that long. They were all under a microsecond, each of these SMD calculations. And the table over here gives you a list of the number of global conformations that I actually visited. And this number is very small. At 4 angstrom separation of the anchors, it's 3,500, and I think that increases to 13,000 conformations at 25 angstroms. The purpose over here is to show you this enormous gap between the conformations that we can imagine, which is 10 to the power 16, and the conformations that we have actually visited, which is really practically nothing. This is what it looks like. But within this hypothesis, this wrong hypothesis of independent dihedrals, we can still use these dihedrals as building blocks. And in principle, we can access 10 to the power 16 structures. And what about the probabilities? Well, since we are assuming these dihedrals to be independent, we just multiply the individual probabilities. And what we can get is this. Pi at delta over here would represent a global conformation, which is given by the 38 values over here. So pi of a probability of a conformation at a given end-to-end distance xi is just obtained by multiplying the local probabilities over here. But all of this calculation is done at a particular anchor separation xi. But the truth is, we don't really care about the tethered molecules. Our real interest is in the free molecule. So the missing link over here is the PMF against the end-to-end distance. Because if we know the probability of a free molecule to have a certain end-to-end distance xi, then we can use that as a weight while we sum up the probabilities of the global conformations at the various sites. So the way to do this is through a separate set of SMD calculations. So this time this is the easiest approach for me. So we set up a set of SMD calculations with constant velocity pulling and stiff springs and Jasinski's equality and so on. The plot that you see here is not the PMF, but rather the probability. And you can see that this probability has a big peak. This is somewhere around 4.5 angstrom separation over here. But by the time it reaches around 10 angstroms, the probability becomes practically negligible. So now I can give you an equation to compute the probability of any conformation delta for a free molecule, not a tethered molecule, but a free molecule. And this is shown by an integral over here. So pi of delta is the probability of a conformation delta, and this is obtained from two parts. The first is P of xi, which is the probability of the molecule being an end-to-end distance xi, multiplied by this conditional probability that I showed you previously, which is the probability of that conformation at a particular xi. But the important thing is, of course, we don't do an integral because we have done SMD calculations only at some specified points over here. Those points are shown by the yellow spheres on this plot over here. So the way we calculate it is through a summation and not an integral. But the achievement over here is that we can now assign probabilities to conformations that were never seen in any of the SMD calculations, right? And in principle, at least, you could give a probability to each of those 10 to the power 16 conformations. Of course, nobody's actually doing that. But now we have to revisit the assumption that all dihedrals are independent, because of course they are not. In reality, we would expect there to be correlations between these dihedrals. And the straightforward way to handle this is to start with the correlation matrix between all pairs of dihedrals at the various anchor separations. So I'm showing 3 of these over here. These matrices are pruned, so you can it shows mainly white over here. So what do we get from this analysis? The most important thing is that we can get communities. Communities are groups of dihedrals that would be highly correlated. And the snapshots at the bottom show us those parts of the peptide that are correlated. Those are shown in colors over here. So as you can see, here are the different communities at the various, from the various SMD calculations, or various stretching conditions. Now, interestingly, it turns out that many of these dihedrals do not seem to be correlated at all. And since I cannot come up with 20 different colors, I'm showing those uncorrelated dihedrals in white color. Now, so we change our assumption. Instead of assuming that dihedrals are independent, we present the independent community hypothesis. So when we talk about local states, we are now talking about community states. Now, but here's the thing. Since the dihedrals within a community are correlated, we can only keep those states that were actually visited, because I'm no longer comfortable about predicting unvisited structures within a community. But when I'm talking about the peptide as a whole, here I can treat the communities as independent units. And here, of course, I'm just free to build even the unvisited structures. The point here is, at this point, we're a little bit more confident about our assertion about the independence of the different parts of the building blocks of the peptide. And when we, this is particularly important because when we calculate the probability of the whole chain conformation, it's now going to be based on the community states. However, yeah, so sorry, this is a new version of the equation. So here we are calculating the probability of a conformation of the molecule at a given xi, and this is built up from the probabilities of the communities. But there's still one problem over here, and which is that the community definition changes across the various stretching conditions. So in this plot, I'm showing you the number of communities detected as a function of the different end-to-end distances xi. Now, although the numbers are similar, but still there's a difference. But more importantly, the membership of the communities are found to change. Now, as the molecule unfolds, it's going to miss out on certain interactions. And these could be like hydrogen bonding, stacking, Sol bridges, and so on. So that means dihedrals that were correlated at some value of sites, at some end-to-end distance, may not be correlated at others. And this poses a problem for us, mathematically at least, if we try to get the probability of a global conformation for free molecule from independent communities at various stretched conformations. That's because a community can either break up or coalesce with other communities. So the way that we're approaching this is by having a consistent definition of community across all separations. So this is what we're calling a segment. A segment would include all dihedrals that are correlated at some point, at some stretching. So now the molecule is built up of independent segments, and the advantage is that I can give you the probability of a segment conformation in the free molecule scenario, but that was not possible when I was dealing only with communities. So the snapshot over here again shows the various segments that were identified in this example problem, and those are shown in different colors. The white color represents there are still some segments that are essentially single dihedrals, and these are shown in white. But we are interested in the larger segments because they're the ones that show more conformational diversity. So first of all, what are the size of these segments? It turns out that these segments are quite small in our example. Many of them are just one-member segments. Some of them have 2 or 3, but the largest one has only 7 members. That is, there are only 7 dihedrals within a segment. Another question over here is, what is the number of states that can be accessed, visited by each segment? And what is the number of states that is possible for each segment? So remember, there's one thing that I I think I have mentioned this previously, that if you carry out a simulation for a few hundred nanoseconds or a few microseconds, you'll never be able to access all the conformations of the full peptide, full molecule over here, but you may be able to access the local states, the states of each dihedral, and even sample them. Maybe not through plain MD, maybe you would require SMD, but you'll still be able to see all the possible states for the dihedrals. So we ask the same questions for a segment now. What is the number of states that were actually visited by the segments, and what's the number of states that can be possible if we assume independent dihedrals? So we did a comparison of both. So the red colored bars over here, so this shows the number of states accessed by all of the segments. This is in log scale, so yeah, many of them I think collapsed to the x-axis at the bottom over here, the single member ones. Okay, so the red color over here shows the number of states that we can theoretically propose for each segment coming from independent dihedrals assumption, and the white colored bars shows the number of states that were actually seen in our SMD calculations, and we see that they're practically the same. So that means we're able to actually see all of the segment states at least. Right. So, okay, so now we have moved over from the independent dihedral assumption to the independent segment assumption, and we can build up the probability of a conformation of the full molecule based on these independent segments. If we choose, we can also focus on local parts. We can focus on the conformations of segments in a free molecule and assign probabilities, and the equation to do that is the same. It has the same parts. There's this P of xi that comes from the probability of the separation of the anchors. There's the other component over here. But now that I have set up over our approach over here, now let us look at some numbers. So let's look at over here. I have two of the larger segments. So segment number 22, this has more than 2,000 possible states, and I have lumped them according to their relative free energies. So yeah, so we can compare the probabilities of the most probable segment state. This is, by the way, probability computed in the free molecule scenario, and the probability of the least probable state. So in the first case, it was 0.098. In the second case, 10 raised to the power minus 14. So 13 orders of magnitude difference in the probabilities. Now, we can, of course, say that the least probable states have very, very big uncertainty. If you carry out plain MD, you'll never be able to see those states, and how can you trust that number? But the point is, now we can predict and actually give some estimate of the probabilities. There's one more thing over here, which is, what is the size of the global conformational space from independent segment over here? So this is something that I've computed at various anchor separations from 4 to 25, and the number of global conformations possible. This is built up because the chain is built up of these independent segments, and I'm counting the number of segment states, multiplying these to obtain the number. So this number ranges from 10 to the power 13 and 10 to 10 to the power 16. And the point is, in principle at least, we can construct all of these 10 to the power 16 states, and I can calculate the probabilities. In principle, of course, that is the point, right? So what do we do?
Sorry to interrupt, Swati. We have about 5 minutes remaining. Thank you.
Right, and I'm almost done. So what do we do with this kind of data? So coming back to the title of the talk, can we predict all of the conformations? Well, we can give an estimate for the size of the conformation states, but it's certainly not practical to generate and scan through all of these 10 to the power 16 conformations. But what we can do is to generate conformations with increasing free energies, starting from the highest probable state So that means we can ask for the top n states. And once generated, we can mine these states for other structural information, collapse onto any collective variable of your choice. So over here, I'm giving you an example. We generated 0.1 million states, 100,000 states in 2 hours on a single processor, rather old desktop over here. So what you can see over here is the explosion in the number of states as we increase the relative free energy. So in principle, we can access maybe tens of millions, maybe even hundreds of millions of such states if you parallelize this. And once you have access to tens of millions of states, you can do whatever you want with it, construct free energy landscapes with as many dimensions as you want, choose whatever coordinates you please to collapse the data. And when I mean generate, I mean actually reconstruct, check for self-intersection, calculate probabilities as well as structural features. So now I come to my final slide over here. So what we have done in this work is we have presented a different kind of framework, based on local states. So we are defining 3 kinds of state spaces for the global conformations. The first is what we're calling the accessed state space, SA. And this is a this has a very, very small size. It's less than 20,000 from all the data that we have. But there's also a constructible state space that is built up based on our assumption of independent segments. And the size of this is huge. This gives us access to about 10 to the power 16 states. And of course, most of these were never visited at all. There's also a third space that is called the universe. And the universe, assuming that we have actually accessed all the local states for each dihedral, then we have already estimated the size of the universe, which is 10 to the power 16. So it turns out that the constructible space, SC, is almost as big as the universe. And so this framework, to summarize, it provides a leap from the accessible to the constructible state space, which of course can have a lot of benefits. So this is still a work in progress, and thank you.
Terrific. Thank you very much, Swati, for your very nice talk. So we have time for a few questions. So maybe I could jump in with the first one, which is to ask how scalable is this method? So you looked at trip cage, but if you were to go to a much larger protein, are you still able to use the same techniques?
Right. I hope to be able to give you an answer in the next meeting. So yeah, this is where we are right now, but that is the whole aim. The point is, even for a large size systems, we may not really be interested in the whole system, but we may be able to apply this in smaller regions and see whether it's possible to use it or not. But I am quite optimistic that we can use it in some format, even for larger size systems.
Perfect, thank you. So we have a question from Anand Srivastata. So please, Anand.
Yeah, hi Swati. Just a quick question. So these methods that exist like ensemble and flexible mechano and things like that, where you take the dihedral angles and each of the phi psi angle you just change a little bit.
Right.
And that would generate you all possible sets of phi psi combinations, right? And
yes,
so that would so how is this different than that in terms of generating the universe of possible, even though non-accessible states?
So right. So if I understand correctly, you're suggesting alternate methods of defining or accessing the local states, and that is something that we are always willing to look at. I just chose what was, to me, one easy way of putting it. I'll have to look at what other methods there are, and the point is how we can connect, how we can get the probabilities and so on. But if there are, there's no hard and fast rule that you have to use only SMD or you have to use only dihedrals. You can come up with alternate definitions of local moves and local states and alternate ways of enhanced sampling. And still, the point is the larger framework, how we are going from this local viewpoint to capture the entire conformational space of the whole molecule. So that is the thing. So, yeah.
Okay, thanks.
Great. And then we had a question in the chat, which I think you implicitly answered from Shivani Verma, which is to say, is there any specific reason to use SMD, steered molecular dynamics? Could we use something else, for example, umbrella sampling? And I think you're saying, yes, you could use whatever sampling technique that you wish.
Right.
Terrific. And so then we have a final question in the chat from Pallab Dutta. He says, nice talk. What would happen at the maximum stretched condition? Would then the number of dihedral angles sort of reduce to some sort of minimal values? So therefore, there's sort of a unique state of the dehedrals at the maximum extension.
Right. So yeah. So you're asking me that when we really stretch it out, what if they all collapse to some unique state? Yeah, then it can all be handled, and that is fine. We will take it. We will take what we see and put it into that probability equation that I showed you. So anyway, when you stretch it maximally, the probability from the PMF that we got, that would be minuscule, really. So those states are highly unlikely to be visited by a free molecule anyway. So that weighting function is going to be very, very small. And if it turns out that all of the dihedrals have unimodal distributions at large scale, stretching, fine, we will take it. We'll take what we see.
Terrific, thank you very much. So with that, I think we're pretty much bang on time. So thank you again to Swati for a nice talk. Please join me in thanking her for joining us today. And now we will have a 10-minute tea break, and we'll reconvene in 10 minutes, and we'll pick up with the second half of the session.
Hello, Frank. So okay? Yeah, you could join.
Hi, now I managed. Yeah, the link didn't work, but I could enter with the. So if I click on the link, it asks me for my email address and a password, and that doesn't work. I mean, then I can. I tried to use the passcode of the meeting there, but that didn't work.
Okay, I see.
So instead, I just went on the Zoom website, joined the meeting with the code.
Okay.
So with the numerical code.
Right.
And with that I got in, but there's some issue with the link, or I don't know what password I'm supposed to use. I don't know.
Thank you. So sorry for that. I will check that.
No problem.
If there's any issue, yeah. Thanks.
How are you guys?
Would you like to try to share your screen?
Sure.
Just deactivate.
So where are we? Here we go. Sorry, that's.
We can see, yes.
Okay.
Hi, Frank.
Hi, Pratish. How are you?
Good. Are you in Berlin or in some beach?
No. I think that was a unique event since we're not doing lockdowns anymore, basically. I'm in Berlin. Yeah. How are you? You just got tenure, right? So that.
Well, it's a long, complicated process. The department voted. Now it goes on.
Yeah. But it's done now.
I stopped worrying about it. Let's put it that way.
Okay. Good. So talk slots are half an hour, right? So we're doing like 25 plus 5?
30 plus 5.
30 plus 5, okay.
Yeah.
Still in NeurOps mode, 25 plus 5 was the standard there.
I mean, it makes even that extra 5 minutes actually makes the talk much nicer. I think 45 starts to become too long. 30 is, I think it's a sweet spot. And we have been getting a lot of question answers. So especially for you, I mean, if you ended up at 25 minutes, I don't think you will have any trouble filling up the remaining 10 minutes just with questions and answers. Because we are getting more questions than we can answer. So we are getting into the chat mode for that.
Okay, good.
We'll make the recording and also the chats available to all the presenters because of the time clash, time zone clash.
Yeah, that makes sense. Yeah.
Some are able to attend the morning session, but some can't make it to the evening sessions.
Yeah, with a meeting like this that's organized between India and the United States, you can basically you could do a 24-hour meeting. Has anyone ever done that? Like a non-stop 24-hour meeting with rolling chairs? I think that would be fun.
Yes, requires a lot of entertainment though. So we have an entertainment session at the end, brainstorming session, which is quite an entertaining session usually in the RARE meetings. So I hope you will be able to join.
And it's led by Alessandro Laio. And I think Laio does a good job of keeping it
Yeah.
entertaining. He goes and provokes, so it'll be great.
So we'll be sharing a set of questions that we have collected from Alessandro, Srabani, and Pratyush, and few others. Jagannath also has contributed. So you can also add some more questions to this brainstorming sessions, and which we will take up in that particular discussion. So I'll share that with everybody today. Pratyush, it's ready. It's ready to share, right? I mean, we can in the current form.
Yeah.
Hello guys, how is everybody? Do you want me to also try share? Not
Yes,
that I expect
yeah,
any
please
surprise.
Yeah, good to see you.
Yeah, I was here, just quiet.
Yes.
It works?
Yeah, perfect. Very nice.
Perfect. Let me stop sharing. No. Okay. I will also go make tea.
Yeah, sure.
Let me try quickly.
Yes, please.
Yeah.
I have made you co-host, I think. Please try. Yes, your co-host.
I'm a co-host now.
Yes, perfect. Yeah, it's coming.
Okay, looking good.
Yes.
Can you hear me?
Screen? Can you go full screen?
Yes. Or not.
Old method.
Just,
Yeah, it's
yeah. Slide transitions are a bit slower.
There is some border to this actually. Maybe is there anything else? Any other option?
Yeah, let's see. You are.
Yes, this is perfect. Yeah.
All right.
Yes.
Then let's do it like this.
Looks great.
Perfect.
Is that a Beamer presentation, Bettina?
That's a Beamer presentation, yes.
Somehow I never switched to Beamer. I should, I guess.
So there are equations, I'm sorry to say. Yep, okay. I'll unshare. There's a stray eye somewhere.
Hey all, just going to pop in and say thanks for organizing this, especially with the multi-time zone.
Hi, Greg. How are
Hey,
you? Nishan,
doing well, and you?
Yeah, fine too. Thanks for joining. You were in both
Wow, my
sessions
pleasure.
very actively. Yeah.
What was that?
I mean, those of you who have not attended the previous RARE meetings, I don't think you can imagine the fun you are missing out we had in the last RARE meetings.
I know.
Like Christoph Delago riding an auto rickshaw with Peter Bullhouse sitting in front at 10 p.m. and going through the city, Agra. It was so much fun.
Michele dancing in a marriage party.
That's correct.
Nice. Yeah, I was sad. I've never been to India before. I've been excited about coming and checking things out.
Next time, I think, in person.
Yeah, indeed.
Greg, you have a very nice book there in the background.
Oh yeah, that's right. Thank you.
That still exists. I need to exhibit that too.
That's right. That's right. And here's the rest of my display.
So it's mostly boost, yeah.
That's right. That's right.
Good. In good shape.
That's right. It was funny. One day, one of the custodial staff came in and he's like, "Hey, dude, I got a question for you." I was like, "Oh, what's up?" He's like, "Oh, did you really drink all that?"
Hi, Frank. Hi, Greg.
Hey.
Hey, Andy.
Great. All right. So we're at 9:50 at least U.S. Central Time. So if Frank, if you're ready to go, I'll introduce you while you go ahead and share your screen. So our next talk comes to us from Frank Noy, who's the interdisciplinary chair in mathematics, physics, and chemistry at the Free Universität Berlin. He's also a visiting researcher at Microsoft Research. And so Frank received his PhD in biophysics from the University of Heidelberg, and he currently leads the AI for Science group as an interdisciplinary research unit active in the development of machine learning methods for physical sciences. So as I think we all know, he works at the intersection of computational statistics, simulation, machine learning, and scientific software development, with broad interests in physics-constrained learning, complex dynamical systems analysis, generative learning methods for statistical mechanics, which I think we'll hear a little bit about today, and accurate methods for quantum mechanics and inference and enhancement of microscopy. And so Frank has very, very many awards, and so I'll just mention a few. He received an ERC starting grant in 2012, a consolidator grant in 2017. He was named the ACS Early Career Awardee in Theoretical Chemistry in 2019, and he was also a Simons Fellow at the Institute for Pure and Applied Mathematics at UCLA in 2019. So without any further ado, please, Frank.
Yes, hi, can you hear me?
Yes, we can hear you well.
Okay, perfect. Thanks, Andy, for the nice introduction. Yeah, IPAM 2019, that was a good time, wasn't it? Before COVID and everything. Great, looking forward to go back to the reunion. Thanks for the introduction. Thanks for putting this conference together, everyone. Very nice job. And so because it's a rare event conference, I thought I'll talk a little bit about Boltzmann generating flows and Boltzmann generators and some of the technical issues that we have been working on the last 2 years. And so ever since we published the first paper, basically we have been working on and struggling with a lot of really, really boring things, like fixing a lot of numerical issues and technical problems, and are basically preparing so that we can essentially scale this methodology to the next level and make it more useful as well. So okay, so here we go. As Andy already said, we are really interested since a couple of years to develop machine learning methods for physics problems, and mostly we're working in statistical physics, but we've also become very interested in quantum chemistry and quantum physics as well. And essentially what's really exciting for us is to try and understand how can we leverage the power of deep learning for actually making progress in these different areas, and what does that mean in terms of how we should design our learning methods so that they can actually give physically reasonable answers or physically valid answers to fundamental physics problems. And so I will be talking about mostly the sampling problem. And so let me just emphasize maybe again why we are so interested and keen on deep learning. And I mean, we've been working with shallow learning actually most of the years before, and also done some work with kernel methods, but it's not what we're focusing on. And the motivation really comes from the fact that not so much whether it's deep or shallow, or whether you can stack many layers and maybe get a quantitatively higher accuracy on some learning task at the price of doing a lot more computation. So that's not really it. So why I think this is so interesting is that you can basically take any program, any numerical program, and reformulate it as a learning problem, because you have this ability to essentially define a computational graph which defines your program, and then because you can differentiate through these operations, every program becomes a learning problem with basically if you have like a deep learning framework to write it in. And that is very exciting because especially if you're working in computational physics, where we are doing this all the time, we're writing computational algorithms, right? So we're writing numerical programs. So now everything becomes learnable, and that can be used as an advantage, of course. So sampling problem. Well, this is a rare event conference, so I don't really need to say much about the sampling problem itself. This is just one of my prototypical examples to show how difficult it is. So this is Barney's bar star, two very small proteins and 100,000 particles in this particular simulation box. And let's say we have some energy function, for example, empirical energy function, and at a given choice of thermodynamic variables, we are also given a probability density that we would like to sample from, such as the canonical density. And doing simulations for this takes us perhaps about a week or a few days on a GPU to get 1 microsecond of simulation time. But a typical binding event is on the order of 50 microseconds on average in the simulation system, which is about a computing year. And a dissociation event would typically take something like 1 million GPU years, which is clearly infeasible even if you have a very, if you have many GPUs and you can somehow parallelize the problem. Okay, so, and we're actually not at the point where we can solve this, what I just showed you with Boltzmann generators, but I think it's still a very interesting exercise to think about a very different sampling algorithm. Because essentially all sampling algorithms that we know otherwise, so pretty much all enhanced sampling algorithms are built upon molecular dynamics and Monte Carlo. And this is simply because these are basically the only approaches we know how to make good proposals for moves in configuration spaces that are so high dimensional. And so basically most enhanced sampling algorithms can somehow be formulated as some sort of biased sampling algorithm, where you change the underlying potential, the Hamiltonian, you change the temperature, which can also be understood as a bias, and so on. And maybe you do umbrella sampling, maybe you do replica exchange, maybe you do metadynamics, etc. So there are many differences in the details, but essentially it's like taking an MDD engine and putting something on top of that, which somehow enhances certain things, and maybe at the expense of other things, like rescue thermodynamics and give up kinetics, for example. Okay, and so the idea here is really that we are trying to move away from that, and we're trying to kind of learn how to make large steps in conformation space. And so the motivation comes from generative machine learning. So in generative machine learning, we are usually given a problem like this. So we're given a set of examples, such as these images of celebrities. And then we are trying to train a machine learning model, for example, a deep neural network that takes random noise variables, so usually uncorrelated random variables as input, and transform these variables into nice images, for example. So basically, we are trying to learn implicitly somehow the probability distribution of images of celebrities, whatever that is. I mean, it's completely ill-defined, obviously. But so we're trying to infer that somehow from the data and then sample from that distribution that we have learned. And surprisingly, this works extremely well, at least for certain data sets. So for example, for images of the celebrities, it actually works amazingly well. So you can really generate faces of celebrities that don't exist, but look very celebrity-like, and do that in HD, in basically 4,000 by 4,000 resolution. And you won't see a flaw with the human eye, at least for most of these samples. Doesn't work equally well for all data sets, of course, but for something like faces, it works extremely well. All right, so in physics, statistical physics specifically, we have a slightly different formulated problem. So we do not necessarily want to learn the distribution from examples, because if we had those examples at the correct distribution, we had already solved the sampling problem. So we may have some samples at a wrong distribution. For example, we may know something about there's a state here, there's a state there, they're relevant somehow, but we don't know their free energy differences because we can't transition between them. And we know the energy, we know the energy, and therefore we also know the probability density we want to sample from, and which is something we don't have in data-based machine learning, where we just have examples. So that's an important difference and an advantage that we can leverage. Okay, so in order to leverage that, so we have a target density. In order to leverage that, we should know from which density we are sampling, because then we can try to match them. Okay, and this leads to a particular machine learning approach. It needs to exact probability methods. So those are methods that allow us to compute the probability that they generate a certain sample. And that's actually a big restriction, because if you think, for example, about generative adversarial networks that are very popular, or variational autoencoders that are very popular as well, both these methods do not have this property. So you can perhaps generate very compelling examples with them, but you cannot compute the probability that they generate that example. But for example, with normalizing flows, you can do it. And that's another deep generative model, which is what we're using here. So a normalizing flow is very often, at least, a neural network. Doesn't have to be a neural network, but typically it is. That is invertible. So we can generate an input Z, push it through the network, get an output X, and then we can also take X and evaluate the network backwards and regenerate Z. And because it is invertible, we can use the transformation of random variables for invertible transformations, which allow us to compute the probability density of the output, given that we have the probability density of the input and the Jacobian determinant of the network. So the Jacobian determinant, that is something we can compute efficiently for certain networks, not for every network, but so basically the trade-off that we have here now is so we have to search a neural network structure that is invertible, so that has some way of inverting also the backward path, which implies that we're not usually changing the dimension on the way. We're keeping the high dimensionality at the input and at the output. And so we have to trade off between how efficiently we can do that forward and backward. Different neural network structures are differently good at that. For example, some neural networks are very efficient in one way, but very slow in the other way. And then it also depends on the neural network architecture, whether evaluating the Jacobian is fast or not. And so depending on what exactly we need, do we need only the forward pass or both the forward and backward pass? And do we need the Jacobian or not? We have to choose a suitable structure. Okay. And in our cases, we usually use everything, forward, backward, and Jacobian. So we take that. We take such a normalizing flow, so such an invertible neural network, and we train it on trying to match target density. And then we're doing basically a statistical physics step, if you like. So we're using the neural network as a Monte Carlo proposal step, and we're doing a Metropolis Hastings acceptance rejection step, or we're just doing importance sampling and we reweight those samples. So either way, we're trying to take the approximate density, which is generated by the network, and then reweight it to something that is hopefully asymptotically unbiased, which comes at a cost. It comes at the cost of a higher variance if we remove the bias, so more noise basically. But in physics, it's usually important that we get something that is asymptotically unbiased. Okay, and that's Boltzmann generator. Basically, the Boltzmann generator is taking a flow to generate an approximate density and reweighting that in some way that we get then an asymptotic, the exact density. And so two things need to be set here. So one, just to maybe reduce the expectations a bit of what we can do with it. First is you do not solve the sampling problem in all of its aspects here. The sampling problem has different facets. And one facet is exploring new states. If you have no idea what states you might want to sample, this method is not going to magically produce them. Not unless you have some internal structure that can, infer other states from states it has already learned, some sort of parameter sharing structure, which we don't really have yet, at least not in an efficient form. But another important aspect of the sampling problem is the mixing problem. So if you know there are certain states here that are relevant, but you don't know what are the free energy differences between them, and it's even hard to construct, for example, a pathway or reaction coordinate or something like that, it would connect these two things because they might just look very different structures. Then this is a very interesting method. So that is really what it is aiming at. So it's best combined with something else that can actually explore states very well, but may just not be very good at finding the correct equilibrium or giving you something unbiased. Okay, so that would be a good combination. All right, how do we train these normalizing flows or Boltzmann generators? So first of all, if we use an invertible neural network, then basically we have such a chain of variable transformations, and each step of the chain is invertible. And we can, for example, start with a certain starting distribution, and that could be something very, very simple, like a multivariate Gaussian distribution, or it could be something that is much closer to the target distribution that we're interested in, such as the same molecular system at a different temperature, or maybe a simplified Hamiltonian, where we are uncoupling certain things that cause sampling problems. So any of these things could serve as an input distribution. Then we push the distribution forward through the series of variable transforms, and eventually we have our target distribution, and we're comparing that to the Boltzmann distribution, for example, if we sample at NVT. Okay, and likewise, we could take samples at the output. So if we have any examples for states that we think are relevant or interesting, that we want to weight with respect to one another, we can push those backwards through the flow, get then basically a corresponding distribution that we want to compare to the prior of the flow, to this mu of z. Okay, so those are basically the two ways we could train the network, forward or backward or both. Every layer here is invertible. That means for every layer, we have this transformation of random variables rule. And we can therefore write down, for example, the Kullback-Leibler divergence, so the KL divergence or the relative entropy between the generated distribution, the distribution generated by the network, and the target distribution. And so it turns out if we do that, we basically get these expressions that you see here. And what that is, if you look at the first expression, is simply you take your input samples, so your, let's say, multivariate Gaussians, you push them through the network does something to them and generates some candidates for molecular structures. Okay. Then you evaluate minus the log probability in your target density. So what is that? That is just the energy. So you just compute the energy of your sample in your whatever MD program or your molecular model that you're using. Okay, so you're minimizing the energy. So far, so good. If you did only that, of course, you would just collapse to some local or global optimum minimum of the energy. So that's not what you want. So the second term, this minus log determinant term, so that is coming from basically aggregating the Jacobians of the neural network transformations throughout the neural network. And what it represents is actually an entropy. So the second term is an entropy term, and overall you're optimizing actually a variational energy, variation-free energy. Okay, so let's look at some simple examples. So here is like just a two-well example where we're basically sampling from the two wells independently, so with basically wrong weights. So we're sampling equally many states in both minima, even though they have very different weights. And then we train the Boltzmann generator forward and backward. Then we sample forward, reweight, and we can reconstruct the correct free energy this way. We can also, as I said, combine this with another method that can explore new states. So here is a very simple implementation of that. We start with one point in state space, and we train a Boltzmann generator on that point, plus a little bit of noise around it. So initially, the Boltzmann generator will just output somehow local configurations around that point. And then we now combine that with Metropolis Monte Carlo steps. Yeah, literally Metropolis, so Gaussian proposals in latent space, so in the Z space, not in X space. Yeah? And then we accept and reject them as usual. We can always compute the acceptance probability of those because we have an invertible transformation. So we can always compute the probability of doing anything in the latent space if we did it in real space. Basically, you always just get an additional Jacobian factor to everything that you're doing, and then you get new points, and you retrain the Boltzmann generator, and with that you can actually explore. And now, whether Metropolis Monte Carlo is a good choice or not, that's arguable, of course. But the general idea here is that you can basically take your favorite sampling algorithm and reformulate it in latent space with this machinery, and then basically leverage the Boltzmann generator's ability to give you whatever samples you generate on the way to give you a better representation of your conformation space by taking essentially all the high probability states and packing them together, or packing them in a way that you can sample them efficiently with your prior distribution. That's reorganizing your probability density in a way that you can efficiently sample it with the sampling approach you're taking, right? So that's really where the power of this approach lies. Okay, so what else can we do with that? So the loss function that I've just shown you is actually already giving you a variational free energy, as I told you. So you can actually directly use that in a fun way. You can do the following. You can take two distinct parts of state space. So let's say you have two different states of a peptide or a protein or something like this that come from two I don't know, X-ray structures or to alpha fold predictions or whatever. And you would like to compute the free energy between them without bothering to construct a reaction coordinate that connects them, because maybe it's a very complicated change and it's not obvious what reaction coordinate would describe it efficiently. And so what you can actually do is you can train 2 Boltzmann generators on these 2 distinct sets of simulations. And so what each Boltzmann generator will do, it will give you just directly via the loss function an estimate for the free energy between your reference state, which is this prior density that you're sampling. So let's say just a Gaussian distribution, and the probability density of that state that you are sampling. And then you can basically do a thermodynamic cycle and just take the difference between those 2, because you're referring to the same reference state, and get a free energy difference estimate from those. And this is very much in the spirit of what solid state physicists do, for example, when they compute free energy differences in materials and they refer to an Einstein crystal, for example, as a reference. Or when you're interested in liquids or gases and you use an ideal gas state as your reference. And so that's the same idea here. Just that the main difference is here you can now learn very, very complicated variable transformations. So you may actually be able to get away with pretty simple reference states, even in complicated systems. Okay, right, so what else can we do? We can, for example, compute certain variables that we are interested in at different temperatures. So here is an example. This is a particle dimer that likes to be either closed or open, but it's in a bath of repulsive particles. They have basically a 1/r to the 12 repulsion. So the system is pretty dense. There's not a lot of space to move things around. So you basically always have to move solvent around when you want to open or close the particles. And so by training the Boltzmann generator, basically just an open and closed states, you can get pretty good free energy profiles here as a function of the distance. And interestingly, you can directly train the system to learn the dependency of these free energy profiles on temperature. And you can do that by basically just varying the width, so the variance of the Gaussian distribution at the input of the Boltzmann generator with the temperature. And so this is not an exact relationship in the flows we are using here, but it can be made an exact relationship with specialized flows. And so this is one of the things we've been working on now. So so here we've kind of made the first step to develop flow architectures that are parameterizable by temperature and potentially also by other thermodynamic variables. And what that means is, so first of all, if you train them at one state point, so at one thermodynamic state point, they would generate a certain probability distribution. And now if you change that temperature dial, this temperature variable or parameter, then the same neural network generates the corresponding probability distribution at a higher or a lower temperature. So it learns the entire family of distributions at different temperatures. And that's, of course, something very nice for physicists and chemists, because then you can do things like melting curves, etc. And you don't need to train independent Boltzmann generators at these different temperatures, because basically you want to learn one family of transformations in one go. Okay, and so otherwise, so what we have shown also is you can generate structures of quite significant systems like this. So for example, this is BPTI in implicit solvent, so about 1,000 atoms. And so you can generate these structures by really sampling from something very simple, a 3,000 dimensional Gaussian, and transforming it. And you get chemically reasonable structures. You don't get like a super good overlap in the energies. You get lower energies, but you're not getting an overlap that is good enough to use this as MCMC proposal steps with this neural network, at least. So basically, mostly what we've been doing in the last 2 years is to look for like methods, flow representations that do that so well that we'll at least be able to do things like this quantitatively. So that we really can do Monte Carlo, we can learn how to do Monte Carlo moves in something like BPDI in implicit solvent and sample quantitatively reliable things. And so a couple of things are needed for this. So one of the aspects that's very important in general is equivariance and symmetries. So like in general in machine learning for physical systems, we often have symmetries in the physical system. For example, in a molecular energy of a molecule in implicit solvent. So that would be invariant to translation and rotation. And so that means the probability density of the Boltzmann generator should also be invariant to those. We should generate a translated or rotated state with the same probability. Or we should simply be not susceptible to this transformation at all in a suitable representation, right? So that's one of the things we've worked on. How can we design normalizing flows? So we built these invertible neural networks such that they have certain symmetries. And so that's one of the things we've worked on. And the second thing, and that's I think going to be extremely important, is smooth flows. So that's just something we just published in NeurIPS this year. And basically, the thing is pretty much all of the existing flow methods, and maybe not only flow methods, but almost all generative machine learning methods are really built to give you a good approximation to a certain probability density. And even if the density is good, it doesn't necessarily mean the forces are good. And so for a normalizing flow, because it gives us a probability density at the output, we can always compute the minus log probability density of an output, and that would give us an energy. And then we can take the gradient of that energy, and that's a force. And we can do MD with it, for example. But in practice, with pretty much all existing flow methods, we cannot do that. It will be absolutely horrible. So we have developed these smooth flows, which basically use smooth functions in certain places in a way so that we can nicely differentiate the energy and get nice forces, and actually even plug these then into MD engines and run them without them blowing up after 5 steps, basically. And so, yeah, I think I'm basically out of time. So I'll just wrap this up. So as just a last comment, one of the things we have worked on is stochastic normalizing flows. Stochastic normalizing flow is basically a generalization of the idea that a flow has to be invertible, an invertible network structure, which is a huge restriction of our expressiveness, and which leads to problems like you see here on top in trying to transform a unimodal into a bimodal distribution and getting this weird connection in between. And we can actually resolve a lot of these problems by introducing stochasticity along the flow. So mixing the invertible neural network layers with things like steps of MD or Langevin dynamics or Monte Carlo. And it turns out that we can actually leverage, and let me just switch to this. We can leverage Crookes fluctuation theorem here, and more specifically an implementation of Crookes fluctuation theorem, which is called non-equilibrium candidate Monte Carlo, turn it into a machine learning problem and use it in this setting. And that teaches us how to train these structures, these not strictly invertible, but statistically reversible neural networks. And then we can actually train them to also act as normalizing flows. And that gives us a lot better sampling capacity and allows us to sample nice smileys or dogs and things like that. Right, and so that's it. Thanks a lot to my group, pre and post-COVID, and thank you for your attention.
Terrific. Thank you so much, Frank, for a beautiful talk. So we have time for a couple of questions. So there's a couple in the chat right here. So actually, we have a question from Mark Tuckerman, which may be related to your latter point about smoothness, which is how does the Jacobian look? And if you have a transformation from a smooth to a rugged mapping, does the ruggedness get shifted into Jacobian, which could cause problems for your sampling? So how do you ensure this doesn't happen?
Could a ruggedness get shifted into the Jacobian? So these transformations we develop here are really smooth functions. So they're infinitely differentiable. So the second derivatives are also fine, if that's your question.
Did that answer your question, Mark?
Mark, you can unmute now.
Thanks. Yeah, I couldn't unmute before.
Yeah,
Hi, Frank. So no, the issue was, so I mean, we played around with transformations at one time too, and so the thing is, so you write down this formula: P of X equals P of Z times a Jacobian.
Yes.
Now P of X is rugged, right? P of Z is smooth. Okay, but the ruggedness is gone. I mean, P of X is still what it is. And so that ruggedness gets shifted to the Jacobian, right? So when you sample Z, you have this Jacobian that's, that fluctuates wildly, I think, right? In order to recapture that ruggedness. So you have to be careful how you sample Z, because you could simply, I think you probably did hint at this with the energy overlap, that, the Jacobian could just obliterate some of those samples because, it's very small in some places, especially where X is a low probability of being, right? But Z may not.
Yeah, so okay, I understand you. So in P of X is whatever it is, right? It's a probability density we want to sample, and unfortunately, it's not very nice usually. And so our problem was that the flow structures, so the neural network structures, introduce additional problems in the sense that they may actually match P of X, but if you take the derivatives of them and compare that to the actual forces or the actual derivatives of the molecular dynamics energy, they would be completely off. So basically they sacrifice smoothness in the forces for getting the distribution right. And so that's what we try to fix. Because if we fix that, then, I mean, I'm not sure how important it is to actually do molecular dynamics on these things. That's just basically an indication that we did an okay job with the forces, that you can do it. But why this is interesting is because now we can also do force matching, right? We can also compare the derivatives from our transformation with the force vector that the molecular dynamics engine gives us for free, basically. And that's a very useful signal because that gives us a lot of information for the training.
Great. So I think we have time for one more quick question from the chat. So I'll just pick one. Demon Ray asks, is it possible to use Boltzmann generators for explicit solvent systems? So
we're working on that now, just for solvent, right? So hybrid systems, no, at this point, not. We definitely want to get there, and it will be a multi-year task, I think. Equivariance is very important for that. So basically what we need is neural network structures that will not change probability density to generate something if we exchange two molecules. And so that, of course, means that we have to give up some expressiveness, but we still want to be sufficiently expressive to express that probability density. And so, yeah, so that's one of the things we're working on right now.
Great, thank you very much. So I think in the interest of time, we should thank Frank for a terrific talk. And Frank, there are a couple other questions in the chat. If you're available to answer those, I'm sure the questioners would appreciate it.
Yep, sure. Thank you, guys.
Terrific. Thank you. So our next talk comes to us from Michele Ceriotti. And so Michele's getting set up, let me introduce him. So Michele is an associate professor in the Laboratory of Computational Science and Modeling at EPFL in Lausanne. Michele received his PhD in physics from ETH Zurich. His interests are in atomic scale modeling of materials, sampling quantum and thermal fluctuations, and using machine learning to predict and rationalize structure property relationships. Michele's won a number of awards. Let me just mention a few. He won the IBM Time Research Fortune Prize in 2010. He was a Volker Hein Young Investigator in 2013. He has an ERC starting grant, and he was also named the IUPAP C10 Young Scientist Award winner in 2018. So Michaela, it's a great pleasure to have you with us today. So please take it away.
Thanks a lot for the very nice introduction and for inviting me to participate. I've been unable to actually take part in any of the in-person RARE events, and I very much hope you will invite me also next time, which hopefully will be in person, because I really miss the experience. So, but still, it's nice to be here, even if just virtually. So, many of you might know that I've been working recently a lot on regression models for predicting the properties of molecules and materials, different kind of properties. But given the subject of this conference, I wanted to focus more on the part of my work that has to do with the description of complex structures by means of automated analysis. And actually, this is a little bit also of a sort of self-critique, trying to look honestly into how the choices that we make in the representations that we use and the automatic data analytics tools that we decided to apply actually introduce effectively a bias into the dimensionality reductions and to the analysis that we perform, and how we can be a little bit more honest about these, getting at the end of the day descriptions that are, in my opinion, more informative. So I don't think there is probably a need anymore to introduce machine learning, not just in this conference, but because all of you probably have heard too much of it over the past couple of years. But, in general terms, I want to reiterate that the basic problem that we are facing is that of taking atomic structures, process them in some form, and then the beauty of it all is that the machine learning techniques themselves are very abstract, and even though, of course, incorporating some degree of domain knowledge into the way you do things, so it's certainly helpful, but in general, the algorithms that we all are using can also be applied in many other domains, and there is a very productive cross-fertilization between different application domains. Now, from the point of view of the tasks that you can perform once you have converted your data into inputs that can be provided as the input variables of a machine learning model, differ, and as sort of anticipated, one thing that you can do is a supervised model that basically tries to associate in one way or another labels, just to stay simple, the interatomic energies to individual configurations. And then you have a number of unsupervised tasks that you can perform that are one way or another related to the search for patterns and regularities into the structure of the inputs themselves without necessarily using any kind of labels. So this operation of looking for recurring patterns is something which is very familiar to chemists, and actually not only theoretical chemists, because at the end of the day, chemical intuition is really the acknowledgement of the fact that in chemistry, both inorganic and organic, you see the same motifs, the same arrangements of atoms appear over and over again. And I'm thinking about, coordination patterns in inorganic materials, chemical groups, if you are a proper organic chemist. And so it's very natural when you have the outcome of atomistic simulation to formalize the process of looking for repeating patterns, for instance, by computing a probability distribution of atomic motifs in some kind of feature space, and then recognize the modes of the distribution and associate them with a kind of objective definition of a recurring pattern. And another kind of task that you can perform which similarly allows you to understand a little bit better the behavior of complicated systems by recognizing some coarse-grained motifs, is dimensionality reduction. Again, in a nutshell, corresponds to taking a as complete as necessary description of the conformations of your system, say the full list of backbone dihedrals if you're working with a protein, and then automatically determine a low-dimensional embedding that you can draw up as a map, and you can understand better what's going on. And of course, this is also incredibly relevant to the problem of sampling of rare events, but I will not touch too much into that. And I will instead focus more on the problem of interpretation. So what these kind of techniques give you is a way to intuitively represent and understand a number of different effects that would be very hard, to comprehend just by enumerating all the possible states. For instance, here, and this is actually not a paper by myself, is this from Mariana Rossi and her group. What you see here in the background are the conformations of different oligopeptides mapped down to 2 dimensions. And in the colored points corresponds to the conformers that are found when these oligopeptides adsorb on a metallic surface. And so by looking at the relative configuration of the points, you can understand in which way the interaction with the metallic surface affects the stability of different conformers in a way that is much more intuitive than if you have to go through hundreds or thousands of different conformers to try and figure out what are the typical motifs. Another example here instead is from my group, and in collaboration with Graham Eday and his team. This was about looking for stacking patterns in molecular crystals, and once again, by looking for clusters, you can rediscover and give a more automated definition the different stacking motifs that are found in the organization of materials that are formed by planar conjugated molecules. Now, what I have to sort of confess is that in both these examples, either as an author, or as somebody who develops the methods and so gives a helping hand to a colleague and a friend, there was a lot of fiddling around with the hyperparameters of the descriptors and with the parameters of the dimensionality reduction to get a representation that was actually really informative. And they actually found this very nice paper, which is about t-SNE, which is kind of a relatively fancy pants nonlinear dimensionality reduction technique. And the paper is really nice, and they have also an online demonstration. And basically, the point is that you can misuse these automated analysis techniques. And for instance, you can take a multivariate Gaussian distribution, run t-SNE, and discover clusters in it, even though the data is actually bona fide random multivariate Gaussian distribution. Or you can get a data set that consists of 2 lines of points, but depending on the parameters of t-SNE, you might end up with something that resembles 2 lines of points, but you can also end up with pretty much anything. So how can we make this process of automated data analytics more honest and less, keep playing around with parameters until it looks nice? So one first thing that we need to understand is the role played by the representation of our inputs. And basically, this, to me, is the main factor that determines the fact that the notion that you can go from a data set to a colorful map that represents the relationship between the different entries in this data set is a complete illusion, because depending on the parameters of the representation, the algorithm that you use, you will end up with a different map. And if you understand your system well enough, actually, this can be an interesting and insightful exercise, but the risk of just fiddling around until you get what you expect is very, very high. And the mapping between actual atomic structures and representations that are used as inputs of your model is, in my opinion, the step in which you are most likely to introduce biases. And when I speak about representation, I speak in very, very abstract terms. So I could be saying I map the Cartesian coordinates to a feature vector, but I might also be speaking about defining a similarity metric or defining a kernel. I really don't care, because actually for a finite data set, you can in practice convert each of these mathematical objects into one another. So it's just a certain way to see the data that you're working with, and understanding how the way you look at your data will determine what happens downstream is extremely important. So, in fact, there are some requirements that you can quite safely enforce onto a representation without really affecting the generality or the flexibility of it. And these are requirements that are essentially dictated by basic physical tenets. So for instance, it's pretty much obvious to expect, but actually not obvious to obtain, that the mapping between Cartesian coordinates and feature space should be injective. So if two structures are different, they should be mapped to a different vector of features, or the distances between these should be greater than 0. And what makes this not so trivial is the simultaneous requirement that if two structures are related to each other by a trivial symmetry operation, translation, rotation, and so on, they should be mapped to the same point, so the distance between them should be 0. And this is, of course, very convenient because it avoids not to have to deal with degeneracies and symmetries and so on. And furthermore, it's typically very useful if your mapping is smooth, because you get better behaved stuff, you can derive it, it's really useful. And something which is very, very also useful in terms of getting models that are transferable is to construct the descriptors for your macroscopic object, your molecule, your crystal, as a collection of descriptors of atom-centered environments. And this is what I'm calling the assumption of additivity or locality. So if you actually look at the past 10, 12 years, the number of distinct frameworks that have been proposed to perform these tasks of mapping Cartesian coordinate into vectors or kernels or distances is huge. And this is from a recent review that we have written, and this is just a small selection of descriptors and features, and basically you can easily find probably several hundreds. And this is, of course, because this is a very important problem, but at the same time, even though most of these have been developed independently, it turns out that the requirements of all of these fundamental symmetries, smoothness, and so on and so forth, actually make all of these frameworks closely related to one another. And over the past couple of years, we have been working on some kind of a unified framework to introduce and construct representations. And the idea is basically to start from the Cartesian coordinates and operate on them in such a way that you progressively make the description more and more symmetric. So a first step involves mapping the Cartesian coordinates that are not invariant to permutations of the labels of the atoms. So, there is a difference in the Cartesian coordinates if I call these carbon 1 or carbon 2, to a density, kind of a field that covers the three-dimensional space in which each atom is associated with a localized function. Let's say a Gaussian, because this is what everybody is using. Now, it's clear that if I give you this field, you can still locate very easily the position of the atoms, but you can't tell me anymore the index that is associated with each atom. So we have made sure that the description is independent on the permutation of the atom labels, which is, of course, something very convenient. And then, you know, this description is still not invariant, for instance, to translations, and you can then obtain instead a description that is translational invariant by building, basically by evaluating the density at 2 points and integrating over translations. This is actually a convolution operation, a correlation to be more specific, and perhaps unsurprisingly, as a result of this operation, you obtain a description for your structure as a sum of atom-centered terms. So basically, the assumption, the ansatz, local and atom-centered ansatz arises as a natural consequence of enforcing translational symmetry over this decorated atom density. And then you can also integrate over rotations. And I mean, I don't have the time to get too much into the details of this theory, but the point is that at the end of the day, you end up with some very abstract objects that are n-point correlations of the atom density that you can generalize to make them equivariant or to also incorporate long-range features. And what is important is that pretty much any descriptor that you can think of can be thought as these abstract objects projected on a suitable basis. For instance, you could think of describing a structure in terms of its X-ray diffraction pattern. Well, this is basically just the 2-point correlation of the density written on a plane wave basis. So it's just a change of basis, and you get a different framework, which of course might be more practical, but fundamentally is the same thing. Similarly, the same descriptors that are used for SOAP, the same objects that enter SOAP, if projected on a specific set of basis functions, give rise to Beller-Parinello symmetry functions. So basically, there is no fundamental difference between SOAP and Beller-Parinello symmetry functions. It's a different way of computing the same object. The different way of computing might have a very different, might lead to very substantial differences in terms of computational scaling, but it's the same object that you're computing. Now, this might lead to the question, then which features should we choose? So in many ways, this is a matter of practical convenience, but we tried to introduce an objective way of comparing the amount of information that is stored in a certain representation. And we do this in a very simple way. We basically take 2 different featurizations of the same objects, the same set of atomic structures, for instance, and we try to see if we can linearly predict feature prime using features as inputs. And the error, the residual error that you get after you tried to linearly encode the feature primed starting from the features F is the global feature space reconstruction error, which is a measure of the residual information that you are missing when you try to reconstruct F prime using all information in F. Now, you can do this in both ways, and the measure is not necessarily symmetric. You can actually end up in different scenarios. You can end up having a small feature reconstruction error in both directions, in which case the 2 feature spaces are basically equivalent. You can end up in a situation in which both errors are large, which means that the featurizations are complementary and encode orthogonal, in a certain sense, the kinds of information, or you can end up in a case in which the error is small in one direction and large in the other, and then you have an objective measure of how much more information feature 1 have compared with feature 2. And this is actually something that here we did for SOAP versus Beller-Parinello symmetry functions, and we find SOAP to be slightly more informative And this is basically a consequence of the fact that SOAP uses an orthogonal basis, whereas traditionally Baylor-Parinello symmetry functions use a non-orthogonal basis, and so you quickly run out of descriptive power. If you computed Baylor-Parinello symmetry functions using orthogonal basis, which of course many people are doing, calling them in different ways, then you obtain something which might even be numerically identically to SOAP. So if these different features are deep down equivalent, does it mean that the maps that you build using them don't depend too much on the parameters that you use in defining the descriptors? Well, actually, no, that's not the case, because most of the dimensionality reduction techniques that we are using take distances seriously. And the distances between structures depend a lot on the emphasis that your choice of descriptors give to different parts of the interatomic correlations that are then discretized to form your SOAP or Baylor-Parinello features. And here I'm giving you as an example different maps that I obtained with kernel PCA, so not very sophisticated technique, using SOAP parameters with different hypers, basically different cutoff radii and different widths of the Gaussians, and deciding to use different atoms as the centers that I use to describe the whole structure. And what you can see here, points are colored based on the cohesive energy, and the shape encodes the number of hydrogen bonds formed by each molecule. And what you can see is that there isn't really much similarity between the different maps, and there isn't a clear correlation between the position on the map and the energy, the stability of different conformers. Now here, have to be honest, we did publish a paper in which we built a map based on this. And basically what we did was playing around with the hyperparameters until we found the descriptors that emphasized the interatomic correlations that were most closely related to the kind of intuitive properties that one might want to recognize. So cohesive energy, number of hydrogen bonds, and so on and so forth. Now, I don't think that this is where we want to get. And so is there a way to basically build these maps without these farting around with hyperparameters until they look good? And what I suggest is that one way of doing it is actually to keep it relatively simple. So no sophisticated nonlinear dimensionality reduction, just PCA, basically your kernel PCA. And so, if you restrict yourself to linear methods or to kernelized methods that essentially are also linear methods in disguise, all that you're doing is basically find linear projections between the feature matrix to a lower dimensional latent space. And then you might also want to do linear regression, another much underrated machine learning technique, in which case you would be basically trying to predict some molecular properties using your features as inputs and doing just a linear projection. And you can also, and this is an interesting exercise, you can do a linear projection using the latent space as inputs. This means that you use only the low-dimensional description of your system to determine the value of the properties that you want to predict. And so it turns out that actually back in 1992, if you look hard enough, you can find a lot of this idea already implemented. People had introduced this principal covariate regression method that quite literally combines a PCA loss, in which basically you're trying to find a low-dimensional projection of your feature matrix that preserves as much as possible the information content in it, basically measured in terms of covariance. So this is really a PCA loss. And this is mixed with a linear regression loss that acts on the latent space. So essentially, we are trying to find a latent space that simultaneously encodes as much as the variance that is present in the original featureization as possible, and simultaneously allows you to regress effectively your target properties. So if you try and run these as a function of the mixing parameter, you basically end up with something that on one end gives you linear regression. You predict your target properties pretty decently, but your low-dimensional map is essentially just projecting the feature matrix on the vector of linear regression weights. So this is usually not particularly informative. In the other extreme, you get PCA. So you get something that emphasizes the features that, based on the representation, on the hyperparameters that you have chosen, is associated with the largest variance. But the ability of predicting, at least in a linear way, the target property is very, very poor. So objectively, if we wanted to understand the structure property relations, this map is very bad because it correlates poorly with the target property. So by mixing the two, you can obtain something that regresses very well and still contains a structure that you can associate with structural features. And actually what we did was just a honestly fairly trivial generalization of this idea by kernelizing the method. And the advantage of kernelizing the method is that actually you get much better predictive power, so you get much better, much more accurate linear regression without actually most of the time major qualitative changes in the shape of the map when you optimize the mixing parameter. Now, how does this work in practice? So this is a typical map. You might have seen something similar in 100 of talks. So this is a two-dimensional map of a data set of hypothetical carbon structures from a high throughput structure search. You could have done the same with the structures of a protein along a long trajectory. This is kernel PCA, and okay, I could interpret this map and convince you that you nicely cluster together structures that are associated with, diamond-like, graphitic, and sp1 kind of carbons. But this is more a posteriori rationalization than something that is actually apparent from the map. If these clusters overlap completely with the background of messy structures that arise from ab initio non-structure search. So if instead you build this map using this kernel principal covariate regression technique and using the cohesive energy as the target, you don't only get a two-dimensional latent space that correlates beautifully with the actual stability of the different structures, but you actually get proper clustering of the different hybridized forms of carbon, and the messy ab initio random structure search configurations end up in a messy blob in the middle, which is, in my opinion, where they should be, and you get absurd simple cubic carbon as an outlier. So this all makes a lot of sense. And actually, if we go back to this problem of looking at these maps of these molecular crystals that would look completely different depending on the hyperparameters that we choose, if we try to incorporate in the mapping the requirement that the latent space should correlate with the cohesive energy, I mean, now the maps start making sense. They do correlate with the energy. We have asked for it, but also they are much less dependent, at least in their qualitative features, from the hyperparameters that we use in defining the representation. And very nicely, we get clustering between structures that have 2 hydrogen bonds per molecule and structures that have just 1 hydrogen bond per molecule. So I'm getting to the end of the talk, and before I conclude, I also want short advertisement for Chemiscope. And this is a tool that we're using a lot to explore these low-dimensional maps. It's basically a portable JavaScript implementation that combines a map and one or more structure visualization panels. So you can click around and visualize the structures that are associated with each point. And this is really very, very useful to, to supplement all of these data analytics techniques with a little bit of click and watch old school analysis. And actually, you have no idea of how many bugs we have found just by analyzing the outcome of a simulation with this kind of interactive tools. We have recently also made it possible to load the same widgets in a Jupyter notebook, so you can also incorporate it in an analysis workflow that uses a Jupyter notebook. So if you want to try it, go on chemiscope.org. And this is a good time also to thank the members of my group that even at 50% presence and masked have been able to work through the past 2 years of pandemic. And it takes a lot of resilience to do that, and I'm really grateful for the hard work they have put in. And so, to summarize the core messages of my presentations is that I've been doing dimensional reduction for quite some time, and it's a great tool to look at the output of your simulations, but it's very easy to abuse it and to read too much into the outcome of particularly of nonlinear dimensionality reduction techniques. And in particular, the representation that you use, so the features that you use to give a high dimensional description of your structures actually can have a huge impact on what's happening. And this is true even when you use representations that are kind of predicated on very general and abstract mathematical grounds, and that in some sense are all equivalent to each other. And a final message is that, as much as deep learning methods are nice, you can also do a lot of nice stuff with linear methods, and the robustness that you get and the possibility of interpreting very easily what you're doing, I think is something which is always worth considering. So thank you for your attention, and there are a few minutes if you want to ask some questions.
Thanks so much, Michele. Please join me thanking Michele for a terrific talk. So we have time for a couple of quick questions. So I could start. Yeah, Michele, I was wondering if you could comment on the similarities or differences between your method and partial least squares regression. It sort of reminds me a little bit of the formalism of that.
So there are they are very similar. So the difference here, as far as I understand, and this is actually not our method. This is really this 1992 P-COV-R method is that you are not, so partial least squares, you basically do the least, because there are a gazillion of very similar methods. So is it the one in which you do PCA and then you do linear regression in the PCA space, or they all have small variations?
Yeah, I think it's one where you sort of do dimensionality reduction on your inputs and your responses, and then you try and fit a model between them in sort of the joint latent space.
So basically, there is a bazillion of methods that are very similar in spirit, and they differ subtly by the way you define the loss. And the reason why I liked this P-COV-R is that it's really defining the loss in the most, in what I find to be the most natural way possible. You really are literally mixing a PCA loss and a linear regression loss. As far as I understand, this is subtly different from principal component regression, partial least squares, but they are all very, very, very similar. And frankly, probably you would get something very similar if you did any of the others. I think it's really just it's more the philosophical idea of stopping to pretend that you can do unsupervised learning and then look for structure property relations in the output of an unsupervised model, if you kind of know a property that correlates with the structural property relation that you want to discover, it does make sense to incorporate a little bit of supervidedness into the map construction. And you don't need to use the end property, but just something that vaguely correlates with it is really helpful to reduce the dependency of the dimensionality reduction on the details of your descriptors.
Great, thank you very much.
And
also
Frank, canonical covariance analysis.
Yeah, the CCA also looks similar. Great. And so maybe I will take one quick question from the chat, which is asking, can you discuss a little bit about what sample properties you could typically use for a real system, for example, a protein?
Oh, I mean, if you have, for instance, if you have some intuitive order parameters, okay, an example of something that I would do if I were to use these to find collective variables, right? So you could take a dozen, interresidue distances or kind of collective variables that you would intuitively use, but you don't know which one is best to use in a given condition. And then you try to find automatically the low-dimensional embedding built on very general and abstract descriptors that correlate simultaneously with many of these descriptors. This might be one strategy to try and find, because I think that what works well, and I mean, this is also in common with principal component regression and all of the other techniques, is that the including basically, the fact that you include in the regression task a component of variance maximization, I think that intuitively this acts as a regularization effectively. So you get something which is not completely, which is not completely flattened out as you would get if you used only the projection on the linear regression weights. And something important that I have to stress is that, one of the advantages of doing this is that this is conceptually different from using the energy as a collective variable, because you can compute these, then, the model allows you to predict these collective variables without computing the full thing. So you could also think of using something which is more time consuming or complicated, and you can just build it in very easily.
Terrific, thank you very much. And actually, thank you to Satya Brata for that question. So great, so in the interest of time, please join me in thanking Michele again for joining us and for a very nice talk. So we'll now move to the final talk of this session from Bettina Keller. And so as Bettina's getting set up, let me introduce her. So Bettina is a professor of theoretical chemistry at the Freie Universität of Berlin. She completed her PhD at ETH Zurich and was actually a postdoc with Frank Noe, who was one of the speakers earlier in the session. Bettina's interests lie in molecular dynamics simulations, QM/MM methods, reaction networks, enhanced sampling, dynamical reweighting, and she's interested in applications to proteins, peptides, riboswitches, and enzymes. And so Bettina's won a large number of awards. So let me just mention a couple of them. So in 2016, she was elected membership to the Jung Academy of the Berlin Brandenburg Academy of Sciences and Humanities, and also a member of the German National Academy of Sciences, Leopoldina. And in 2018, she won the Hans G.A. Hellman Prize for Theoretical Chemistry. So we're very pleased that Bettina can join us today. And please, Bettina.
Thank you very much for this kind introduction. Thank you for including me in this session or in this conference at all, and for putting this together across 3 different time zones. I really enjoyed the afternoon, at least the afternoon in Europe, and I don't know what time zones you are in. And what I brought you today is something a bit about dynamical reweighting and path probability ratios to sample rare events. So rare events. Well, yeah. That hardly needs an introduction, right? So if you think of molecular dynamics, these molecular dynamics occur across relatively narrow transition paths. You can think of this like mountain paths across mountainous energy landscape. And even though these paths are narrow, right, so they don't cover the entire space, we still have huge barriers along these paths. And we can speed up the simulations, of course, by biasing these potential energy function, and for example, using something like metadynamics. And then we explore the state space faster. But the drawback is, of course, because we speed up the rates, the dynamics get distorted. And there are lots of methods that recover the stationary properties, but we would also be interested in recovering the dynamic properties. And methods that try to do that are called dynamic reweighting. And there has been quite some effort in developing dynamic reweighting methods. So there are different ideas. We tried to classify some of them. There are actually so many that we couldn't classify all of them in this short review over here. So one idea would be to rescale the transition flux across a state in the conformational space. So to have if you start here in this cell and then you transition out into the neighboring cell, the transition probability will be different depending on whether you simulate at a blue potential or at a green potential. So blue is going to be the simulation potential, and green is going to be the target potential throughout the talk. And then there are other methods that try to reweight the overall transition probability from one state I to another state J. Reweight the Markov model, essentially, often based on likelihood functions. Trum and Dum are examples. And we've been focusing on methods that kind of reduce this or decompose this overall transition into individual paths that make their way from I to J. So instead of reweighting the entire transition, we reweight individual paths, and that's called path reweighting or Gilsanov reweighting. And as I just said, I want to focus on path reweighting today. And one reason for this is that this is actually, or in principle, an exact method. So you don't make any assumption in the reweighting process. In principle. And what I will talk about today is this in principle. You see, the problem is that, and that is not only in path reweighting, that applies to many analysis techniques that we have. Is we tend to simulate Langevin dynamics. So we use a Langevin thermostat that has a friction term and a random force to represent a thermal bath. But what we analyze is often based on overdamped Langevin dynamics. And a lot of theory has been developed for overdamped Langevin dynamics and not so much for Langevin dynamics. And in particular for path reweighting, we have a lot of equations or a lot of theory for overdamped and for underdamped Langevin dynamics. This is lacking. This is because overdamped Langevin dynamics is a first order stochastic differential equation, right? So you have a first order derivative over here, first one derivative with respect to time, whereas this is a second order stochastic differential equation where you have a second order derivative with respect to time, so an acceleration, and that seems to make things very, very complicated for the mathematicians. I don't know. So we're going to look at this discrepancy between overdamped dynamics and underdamped Langevin dynamics. But let me introduce path reweighting first. So what we would be interested in is reweighting a dynamical property, and often this is some sort of a correlation function. And as an example, I pick here the transition probability, or the overall probability of observing a transition from I to J. And you can formulate this transition probability, Cij, as a path integral, where you integrate over paths of length tau, so of a certain length. All the paths have the same length, and you evaluate the initial point and the final point with a indicator function. So if the path starts in I and ends in J, then this makes a contribution to the path integral. If you then discretize this for discrete paths, this looks like this. Here, the blue is your simulation, right? It's what you simulated, little snippets of your simulations of length tau. that have an initial point, and then after n time step have a final point. And you can really imagine that you have a long simulation that you snip down into little pieces. And if they start in I and if they end in I, then this evaluates to 1. So this little path makes a contribution of 1 to the overall count of transitions that you've seen in your simulation. And then you divide by the number of paths that you have in your simulation. And blue means that the path has been generated at the simulation potential. And here in our case, this is usually a normal potential V tilde plus some bias U of X. Now path reweighting. So this is what you would normally do if you simulate, and then you count the number of transitions and you get a rate or a transition probability. Now in path reweighting, you would like to do that. You would like to estimate the rates of your target potential by analyzing simulations conducted at the biased simulation potential. You would like to have the rates of the green potential by looking at trajectories sampled at the blue potential. And of course, the equations in principle look the same as previously, only that you now have to introduce a different probability density over here. So here we had the path probability for the simulation potential, and now we have the path probability for the target potential. And formally, we can rewrite this, or we can decompose this into a product of the original, the blue path probability, times a reweighting factor. And if we discretize this, what happens is that every path sniplet from x0 to xn that starts in I and ends up in j makes a contribution to the path interval, but now not with 1, but only with this relative probability here in red. So in order to estimate the correlation function at the target potential, we need to sum over all paths in our simulation, check whether they start in I and end in j, and then we need to calculate this relative weight. And we decompose the weight into 2 terms. One is the probability of finding the system in the initial state, and the other is the probability, the actual path probability of seeing the path given that the state has started in x0. So the probability of the path omega given that we started in x0. And these are just ratios of the relative path probabilities in the target potential and the simulation potential. So our goal now is to calculate these factors. And the G factor is actually simple. The G factor is just the Boltzmann factor of the bias. If you divide the Boltzmann probability densities, you end up with this Boltzmann factor, which contains the bias over here. And next we need to calculate, or we need to come up with a model for the path probability ratio. So we need to calculate the probability of observing a little path given that the path has started in x0. And this is done by decomposing the path into the individual integration time steps. So if you have n integration time steps, then each of these dots is the progress in your simulation at every integration time step. And then the overall path probability is the probability of observing these individual steps. So the overall path probability is just the product of observing these individual steps. And so there's no there's no Markov assumption from going here to here. We only have the Markov assumption at the integrator level, and this is Markovian by construction. The problem is that now the single step transition probability, this one over here, going from xk minus 1 to xk, that of course depends on the integration scheme. So here we need to insert knowledge about the integration scheme, and that's where the difference between overdamped and underdamped Langevin dynamics comes in. For overdamped Langevin dynamics, these equations are well known, and you don't need to go through them. I'm just saying that the slide is supposed to tell you that we all know them, right? So that they have been well documented. So this is the typical integration scheme for an overdamped Langevin dynamics. That's Euler-Maruyama, and we have the single-step probability that looks like this. And if we take the ratio of single-step probabilities with target potential and simulation potential, we end up with a probability ratio M that contains here the difference of the gradients and the difference of the gradient squared. So this is well known. And if you simulate at overdamped dynamics, this really works like a charm. So what we did here is we simulated at this double well potential, and we reweighted to the triple well potential. And the simulation was conducted with overdamped Langevin dynamics. And you see, we're not terribly squeamish with distorting our potential. Yeah? We make it smaller and we make it tighter, and we introduce additional barriers where there used to be additional barriers and minima where there used to be maxima. So you can actually do quite a bit with the potential without breaking the method. And what we use to benchmark the system is the Markov state model eigenvectors. So this is the first one is the Boltzmann distribution, and the second one represents the exchange between this minimum and this minimum over here across the central minimum, and the second one represents the exchange between the central minimum and the ones on the side. And then we have timescales associated to this. So that's 2000 time steps and 600 time steps. And black is the reference simulation where we actually simulated in the black potential, in the triple well potential, and red is the reweighted trajectory. So we simulated in a double well potential and reweighted to the triple well potential. And for overdamped, so if you conduct our simulations with overdamped dynamics and we reweight with overdamped dynamics, this is right spot on. Now but if you want to simulate a molecule, you of course turn on Langevin dynamics, underdamped Langevin dynamics. And one idea would be, well, if we have all these equations, we just use them, right? We ignore the problem and we use it. But this unfortunately fails very badly. So we simulated at the double well potential using underdamped dynamics and reweighted to the triple well potential using the known equations. And what happens is that not even the eigenvectors are recovered. So even the Boltzmann distribution is badly distorted. So you get very wrong results if you just use these overdamped Langevin equations, overdamped Langevin reweighting factors. And what we had to do now is to develop a reweighting factor for underdamped dynamics. Okay, and that's in this paper. So we used a very simple Langevin integrator to wrap our heads around this, to understand how the method works. So that we call it ISP scheme, and it's actually an integrator that we simplified, right? So it's not an actual integrator, right? So you won't find that in an MD program. It's a very simplified thing. But it is the advantage of this is that you can actually kind of see the basic things that's happening. And the basic idea here is that an integrator is actually a transformation from a random number to a position. So here's the update in the position space, and it takes the initial position and it takes the potential. And most importantly, it takes the random number. So this is a very complicated function that transforms a random number into a position. So you can think of an MD integrator, and that is true for all Langevin integrators. Well, for all stochastic integrators, you can think of them as a transformation from random numbers to positions, and vice versa. So because also you can also invert this transformation. So there's a direct mapping between random numbers and positions. And the upshot of this is that you can replace your path probability by the probability to draw this string of random numbers. And the probability to draw the string of random numbers is of course known, because we know that this is, we construct them to be standard Gaussian random numbers drawn from a standard normal distribution. with a probability like this. So given the random numbers, we can very easily calculate the path probability like this. Okay, so here's the idea. So same thing in pictures, so to say, right? So in an MD simulation, what you do is you generate a sequence of random numbers. You have an initial point, you have your integrator equations, and that's kind of a black box, and it returns positions. And now if you want to calculate the path probability, you reverse this process. You take your positions and you reverse the equations, and you get out the sequence of random numbers that you would have needed to generate this path, and you can calculate very easily the random number probability, or which is then equal to the path probability. And you would do the same thing for a different potential. So you would take the path that you have generated, and then now you would like to invert the integrator equation for a different potential. You get out a different sequence random numbers, and then this is the probability of drawing these random number sequence for the target potentials. And then by comparing these two probabilities, you can get to a relative path probability. So we can look at this like this as well. So here's the path that you have generated, and you would like to reweight this. You have generated this as at the simulation potential, and you would like to reweight this to the target potential. Here in solid, we have the random numbers that are needed to generate the path at the simulation potential. Then here, the dotted lines are the random numbers that are needed to generate the path at the target potential. And if you reweight, or if you take the ratio of these two probabilities, you can get the path probability ratio M that you would need to reweight your transition counts. Now, the nice thing is now that we can formulate the random numbers at the target potential as well, the original random numbers eta k plus a little delta, plus a little shift here. So we can get to the dotted line by taking the solid blue line and adding this little shift to the target random numbers. And if we do that, if we construct or we write down our tilde eta k like this and insert this into this equation, we end up with a formula for the path probability ratio that looks like this, because that's a binomial formula over here. So here we have a binomial formula, and then the first term cancels, and you're left with these two terms. So why is this useful? This is useful because this is the random number for the simulation at the simulation potential, and that is something that you don't have to calculate. That is something that you can just record during your simulation, and the only thing that you need to calculate is this little orange shift. So the idea now is that in order to calculate this path probability ratio, you record the random numbers of your biased simulation eta k, you calculate delta eta k, and you evaluate this function. And for the ISP scheme, delta eta k looks like this. We have here the gradient of the bias, and we have here a factor xi delta t, that's the friction times the time step. And we have here a prefactor that depends on the temperature. So this is exact, and it's much easier and less error prone to implement than calculating the full path probability in terms of positions. We also derive the full path probability, but this is really a much cleaner and simpler formulation. So with this, we can now do exact reweighting for this ISP scheme. But the story goes a bit further, because if you look at this and compared, so this is the expression for delta eta k for the Langevin integration scheme, and that's the one for the Euler-Majorana scheme. And what immediately comes to mind is that they look very, very similar. So we have the gradient of the bias over here. We have this prefactor that contains the temperature and the mass. And then we have here a factor that essentially evaluates to 1 because Xi delta T is very small for MD simulations. And we have analytical proof that these two factors, they're really, really similar up to 4th order, right? So up to 4th order, you can replace this factor by this factor. So what you could also do is you could record your random numbers at your simulation potential during the simulation. And then the correction for the reweighting, you can just replace by this Euler-Majorana scheme at delta eta k. So here's the error analysis between the exact expression and the approximated expression. So the difference between ISP and Euler-Majorana, difference between underdamped and overdamped dynamics. And as a function of the time step, essentially, MD simulations are conducted here in this regime, and you see they're right on top of each other. And this is the absolute error in delta eta k, and that's the relative error. It's really 0. But what is important to point out is that so if you have an update in the position from xk to xk plus 1, this is composed of a deterministic update like this, and then a random update like this. And that is very different for the different dynamics. So this, if green is Langevin dynamics and blue is overdamped Langevin dynamics, this might very well look like this. We have very different, the overall progress is composed of very different sub progresses. But what is similar is the sensitivity of the random number on the change of the potential. So this delta eta k is the same for underdamped and overdamped Langevin dynamics. So the delta eta k represents how much does the random number change when I change the potential. And that seems to be the same for both dynamics. Okay, so let's try this. So that's our model potential. We simulate at the double well potential using Langevin dynamics. Black is the solution when the reference solution when we actually simulate at the target potential, and in green we have the reweighted results from the double well to the triple well potential. So that works well. And in orange, we have the reweighting where we've replaced the delta eta by the Euler-Majorana expression, and that also works very well. So that's right on top of each other. So obviously this approximation works very well for the ISP scheme. And for the implied timescales over here, actually the approximation seems to work even a bit better than the analytical result. And the reason we think is in numerics, because we have to calculate less exponentials here in this reweighting factor than in the approximation. So the approximation has to calculate less exponentials than the actual analytical exact expression. So we think that this might be the reason why the implied timescale of the approximation are actually a bit better. So now we have what we have achieved now is a method to reweight Langevin dynamics, and that in fact works also for molecules. So what I show you here is a beta hairpin peptide that if you simulate this in an unbiased simulation, we have here 17 microseconds. The only thing that you see is a slight opening and closing of the ends. And these are the two states that you would see in an unbiased simulation, open and closed. And they do that on a time scale of a nanosecond. And but we know from experiment, well, it's been designed like this, that this is a molecule that open and closes. It's a molecule that has been designed to open, unfold, and refold. So we biased along these three of these hydrogen bonds, so three one-dimensional biases, and in fact saw full opening and closing of this peptide. And then we reweighted this and obtained a reweighted Markov model that has three states, A, B, and C, where B is the fully closed, and we have transitions to half open states left and right. And a state that was sampled, a fully extended open state that was sampled, got reweighted to very low populations over there. So this works. And we have 1.4 microseconds of simulations to conduct this. So an order of magnitude of simulations less than previously. And if I have, and I'm not quite sure how I'm doing time-wise, I have a little,
yeah, you have about 5 minutes left. We started a little late.
Oh, excellent, excellent. So what I've shown you here is now something that works for a very specialized integrator, right? So for this ISP integrator that we constructed, because that was what we could handle. Of course, we would like to generalize this to actual and large event integrators, right? So actual large event thermostats that you would use in your simulation. And that is what we're currently working on. And I show you how to do that in principle, right? So how you would derive this random number difference for an actual large event integrator. What I show you is the ABOBA method. That's one of the splitting operators, splitting algorithms that you might know from works of Ben Leimkuhler, Gavin Brooks, or John Codera. And they now have 6 steps, A, B, O, B, and A. And the way to derive delta eta k for this one, right? So there's one random number drawn somewhere in the middle, and you ask the question, what random number should I draw in order to get the same step with a different potential, where the potential is evaluated at 2 positions. So that's a bit more complicated. And the way we do that is we write down the initial point, QK and PK, in this phase space diagram. So it's a phase space over here. So that's the initial point. And after one integration time step, we want to get to this final point over here, QK plus 1, PK plus 1. And the integrator breaks this transition down into 5 steps. A, an update in the positions, B, O, B, and 3 updates in the momenta, and then A again, an update in the positions. Now, these vertical lines, they don't depend on the potential whatsoever. So they will be the same whether you conduct this at V or at the target potential, at the simulation potential, the target potential. What changes are these B terms, because they depend on the potential over here and over here. But in order to get the same progress from K plus 1, we require that this distance remains the same. So if gray is your simulation, so the decomposition, the simulation potential B1 O plus B2, then in the reweighted simulation B2 and B1 might have changed, and then O needs to compensate this change in distance such that you have the same distance over here. So these 3 terms need to be the same as these 3 terms in the reweighted system. Yeah? And the updates, the distances you get from these 3 equations in here, and then you change the potential and solve for delta eta k. And what you get out is something that looks like this. And this again looks very close to the Euler-Majorana expression, the overdamped expression. And we tried that for a few large event integrators, and it seems to work really, really well. So here is the reweighting for the ABOBA scheme. Again, our triple well potential, and the eigenvectors are right on top of each other. So that's a direct simulation. That is the reweighting with ABOBA, where we use this equation. And that's the reweighting with the approximation, yeah, where we use the overdamped large event equation. And we also tried a stochastic position valet, and that also seems to work very well. So this approximation seems to hold up for many large event integrators. What this means is that maybe, probably, that is something that we're looking at right now, you don't have to derive this delta k for each integrator separately, but you can kind of import the large event integration expression, right? So then a general path probability for large event dynamics would look like this. You record your random number at the simulation potential, and that is easy. You can just write that out into a file. You also need to record the bias at each integration time step. And then you evaluate delta eta k using this this expression, and that seems to be universal from what we see currently. And that would mean that path reweighting becomes actually technically very tractable using MD programs like OpenMM, where this is easy to implement. It would become something that is relatively simple to integrate, because the only thing that you need to evaluate is this equation. So with this I close, and I would like to thank my group for working through this pandemic. What I showed you today is work from Luca Donati, who started the path reweighting project, and in particular Stephanie Kieninger's work, who did the exact and approximate path probability ratios for Landau dynamic. And then the last thing that I showed you right now is something that we're doing together with Simon Giesbrecht and Stephanie Kieninger. And we started a project together with Carsten Hartmann from BTU Corpus, and funding, and you for your attention. I'm looking forward to questions.
Great, thank you very much, Bettina, for a wonderful talk. So we already have a couple of questions in the chat, and so a question from Peter Ballhaus. How does the path reweighting scale with path length? As the path becomes longer, do we not have exponentially more unlikely reweighting factors? And how does this translate into the rates?
Yeah. So I kicked out the frequently asked question slide. So yes, indeed, it scales. So the path probability goes down exponentially with path length. So if you pick a path length that is as long as the transition across a barrier, it's probably not going to work. So what we do is so if you think of these transitions, these going from I to J, these are really Markov model. So that the length of these paths is the length of the lag time in a Markov model. So orders of magnitude smaller than the slowest time scale in the system. So the combination of Markov models and path reweighting seems to work.
Great, thank you. So we have another question. Can you provide more details on our reweighting 3 one-dimensional method dynamics bias used in the beta hairpin system? That's from Angie Babu Kapakalia.
What was the last one?
Can you provide more details on how you applied the 3 one-dimensional method dynamics biases for the beta hairpin system?
So well, we constructed 3 one-dimensional biases, metadynamics biases using Plume. So that's not we didn't sample the whole cube, not the 3-dimensional system, but we had like 3 one-dimensional biases. And then to reweight this, what you need to have is the gradient of the bias with respect to, so where do I show you this? Here. So what goes in here is the gradient of the bias, but the bias now only depends on these 3 distances, right? So you only need the derivatives with respect to these distances. So if the bias is low dimensional, then also this gradient is going to be relatively low dimensional. So in R, it's 3 one-dimensional gradients, and then you need to translate this into Cartesian coordinates. Does that?
Great, thank you. So I had a question, but it may have been on your FAQ slide, which was the degree to which you can apply a bias. I think that you recommend not applying.
We don't know that actually. So sorry, we don't know that. How strong should the bias be? It's very difficult to analyze, right? So you can test that, of course, for many, many different systems, right? But to have a general recommendation is very, very hard to say, right? So what we find is the uncertainties become too large if you use the entire, like the fully converged metadynamic surface. What we do instead is we take the fully converged metadynamics surface and multiply or divide by 10. Yeah, so we take 10% of the full system, and that worked well for the systems that we looked at. But so what I maybe would also show. So when should you use it is maybe the more more relevant question, or the one that I can answer at least, right? Is so what we did here is we have butane at 3 different temperatures, 300, 200, and 150. And so if so in all cases, we recover the correct eigenvectors by reweighting. By biasing and reweighting. At 300 Kelvin, this doesn't really give you a gain, especially at at larger implied time, larger path lengths, then the uncertainties become large. So essentially, if you have a system where you can sample the Boltzmann distribution without bias, right? Even if you can wait long enough to get the Boltzmann distribution sampled, then you should use this. You should use direct sampling. But if you go to temperatures or barriers that you cannot cross with direct simulations, so here the direct simulation yields errors in the Boltzmann distribution and then large errors in the second and third eigenvectors. Their reweighting actually gives you an advantage. Gets the eigenvectors, gets you the correct eigenvectors, and gets you at least sort of good estimates for the implied timescales. The slowest implied timescale is not so. Sorry, that is actually, we think that is a wrong estimate, to be honest. So that's the direct estimate, but the direct estimate didn't converge the Boltzmann distribution, so that is probably a wrong estimate. So this is a method to be used when you cannot sample directly. I think then this is
great. Thank you. And then we have maybe time for one final question from the chat from Anmol Kumar, who asks, how does the time step of the simulation influence the path probability?
Good question. Yes. So you're a bit limited with the time step because you still have I mean, you would need to constrain so you're limited to 2 femtoseconds because of the potential energy function. That no reweighting method is going to change this, right? So if you don't constrain bond length and angles, you cannot increase your time step. But suppose you could increase your time step, right? Then let's see whether I have a good here. So here we plot the error as a function of xi delta t. Xi is the friction, or sometimes this is called, in chromics, this is called the time scale of the integrator. And it's often set to something like 1 over picoseconds. So an order of magnitude larger than the implied time scale, than the time step. So if you set the time step to 2 femtoseconds, you're running your simulation in this regime. If you increase the time step and keep the friction the same, then eventually you will get into a regime where this is not accurate anymore, right? So where where both the integrator and the reweighting is not accurate anymore. But this sets in really at 100 femtosecond timescale. So it's for a long range, these are actually. So we're not, so at least until 10 femtoseconds, I think the larger one integrators are stable, and the reweighting is stable until 100 femtoseconds, supposing that you could simulate at 100 femtoseconds. So there's a huge margin until you run into any accuracy errors with the time step, because so far we're limited to 2 femtoseconds, or maybe 4.
Great, thank you very much. And so, so please join me in thanking Bettina again for a wonderful talk, and thank you to all the presenters in this session. And so, so the talks have been recorded, and we will post the videos on the RARE webpage, and we will pick up again for the second day, beginning at 8 a.m. Indian Standard Time on December 16th, and that will be chaired by Arnav Mukherjee. And then Nisanth, do you have any final announcements before we wrap for this session?
So please join the poster sessions, encourage the young scientists there. So the poster session, those who have registered for the conference, I have sent an email on how to log into the platform. So all the posters are very exciting, so please join, put your comments in the chat box. And on the 16th, that is tomorrow night, Indian time, we have a poster session where you will also see the poster presenters live there, so you can interact with them. And so we hope that we can make it very lively. Thank you so much. Thank you, Andrew, for chairing the session. It was really great. Thank you.
Thanks, everyone. See you tomorrow.
Thank you. Bye-bye.
Have a great day, and good night and good morning.